Good morning and welcome back to our ninth annual RRP Scientific Conference, day two. We had a great session yesterday with a diverse group of speakers, several not part of our RUNX1 FPD community, but today is really a day full of RUNX1 FPD only talks. So different from yesterday, and I'm really looking forward to hearing all these fantastic presentations that are lined up for the day. I'm just going to quickly go through the announcements I went through yesterday because if some of you weren't there at the very beginning, you may have missed it. So, and for those of you who have to listen to it again, apologies, but I'll go quickly. So, please register. We are going to have our in-person conference in May. We'll be in Atlanta, Georgia. I think it should be a really beautiful time of year there at the Emory Conference Center Hotel. And the scientific session will be the May 6th and 7th. And then the patient meeting will actually overlap with us May 7th and 8th. We'll also, those of you on the SAB, will also have an SAB meeting on the 8th as well. So please, if you haven't registered already, please do so. Next, here we go. If you have colleagues who are not familiar with hereditary heme malignancies or maybe don't believe that they're all that rare, please send them to this webinar. So we continue to do medical education webinar series, and we continue to be surprised that we hear there are still groups out there that don't think about germline testing. So this is an effort to demonstrate and report out on all the literature now showing that these types of malignancies are not all that rare. So Dr. Bolton will be giving a presentation, and she actually goes over some really great research data from her lab analyzing the UK Biobank and the risk of germline variants alongside CHIP and how that CHIP and a germline variant increases risk quite dramatically. And she'll be joined by Dr. Rangaraju as well as Kelly Smith-Simmer, a genetic counselor, in a panel discussion afterwards. So please help us spread the word. We have a new, well, it's not a new, it's a biannual rare grant that we host every year or every other year with the ALSF. And please look at the RFA. It's available, and the deadline is December 11th. We hope that you consider applying. It's up to $125,000 per year for 2 years. In addition, we have another very large grant mechanism coming available next year. The due date is June 1st, 2026. This program is in collaboration with the American Cancer Society. It's called our LEAP grant and offers $215,000 per year for 4 years for an individual investigator, and then $430,000 per year for 2 years for a team investigator or team group. So please check it out. This is just to let you know that we do have resources available to patients and families to help them learn about the different research opportunities available. There's a QR code here, and you can find information on our website. Also note that we recently launched our RUNX1 Patient Data Hub, our patient tissue bank, so that we can help support the research that's ongoing in this community. And then I'm just going to end with the virtual patient meeting. So we have a virtual patient meeting next week. Please, you're welcome to join, but also share it with your patients and families as well. And I think I'm ending there. Oh, really quickly, I think we talked about this yesterday, but just make sure that you have your microphone off if you're not speaking. Use the raise hand for Q&A. And then we are recording this only for internal purposes, and these presentations will not be shared broadly. However, if there's someone who reaches out and says, oh, I really wanted to see John McClatchy's talk and I missed it, then I would reach out to John McClatchy, Dr. McClatchy, and say, is it okay if we share with blah, blah, blah. And timekeeping, Lydia Harper will be jumping in and giving everyone the 1-minute warning so that we can keep this agenda going. Okay, I told you I'd be fast, and I got there. Okay, so we're going to start with the young investigators today. These are individuals who applied with an abstract submission and were selected to present today. So Dr. John McClatchy will kick us off from Oregon Health and Science University. He works in Dr. Anupriya Agarwal's lab. John, please take it away.
Okay, hopefully people should see PowerPoint slides and they should hear my voice.
Yes, you're good.
Those are two great starts. Let me turn on the laser pointer. Okay, man, people call me doctor now. That's new. Today I'm excited to talk to you about mutation acquisition within the RUNX1 background and the mechanisms that expand this. But first, some rationale. This is a slide no one's seen before, but we have clonal expansion occurring at a much accelerated rate inside the germline RUNX background, almost tenfold as high. And when we look at the mutations that are predominantly expanding within this background, we see that they are a little different than those that occur in a non-FPD background. They have higher B core, they have higher TET2, they have much lower DMT3A. And our work today is focused on TET2, and we've coordinated with the spec lab. They are working more on B core. And so together we're kind of exploring some of these mutations expansions. TET2 is our specific target. Now, the RUNX1 background, as many of you are aware, is considered very pro-inflammatory. This is data generated from Mona, who many of you will be familiar with from our laboratory. And this is bone marrow cytokine data from aspirates, from patients, and it's just showing that many cytokines are upregulated. And this is of interest because clonal expansion, of course, occurs in an accelerated rate over age. And inflammatory cytokines have been shown to coincide with this and in some cases drive it, such as the cases of IL-1, TNF-alpha, and LPS have all been shown to have effects that help promote the expansion of these cells. And this kind of leads to what started as my central hypothesis, that we would have these germline RUNX1s. Germline RUNX1 has higher inflammation. Inflammation drives TET2 expansion, and that expansion usually occurs from long-term hematopoietic stem cells. So it's a fairly logical starting point, I felt, at least. It could be wrong. But we set out with a few objectives to make a mirroring model of this expansion using this RUNX1 R188Q mice from Lucio. To characterize alterations in hematopoiesis to see if the germline RUNX1 setting had any specific host effects that were driving this expansion more so than would occur if the non-hematopoietic cells were also wild type, and then to see what mechanisms and therapeutic targets could be exploited to stop this expansion. Starting on this first objective, I use for most of this work, I've used a few models, but I used a transplantation model. This is, once again, the RUNX1 R188Q mutant mice with the RUNX1 TET2 mice, floxed in this case, competing, induced from an SCL-CRE, and transplanted into CD45.1/2. These are in the RUNX1 background for the recipients as well. And I've done two different transplants into this. One is still ongoing. That's the one on the right. One was a 1-to-1 ratio starting. The other was about a 1-to-3, 1-to-4. And in both cases, this dark purple line has shown fairly clear expansion over the course of time. And I've confirmed this by other methods too. And cumulatively, what this suggests is what you would expect. RUNX1 TET2 outcompetes RUNX1. And it does so in this mirroring model, which is maybe a little more important because we know it happens in patients. Otherwise, why would it be so prevalent? But does it happen in our models? Otherwise, they're not very good models. Importantly, we also see this occurring from the top of the hematopoietic hierarchy, from the long-term hematopoietic stem cell population. And I next wanted to look at, okay, we see these differences and we see them in the top of hematopoiesis. How are they altering the direction of hematopoiesis? Now, TET2 is historically myeloid biasing outside of the RUNX1 setting. And RUNX1 can also be myeloid biasing. Is it still myeloid biasing? So brief explanation of these graphs, because there's a lot of populations, and it's a little hard to put them all on one panel and have them make any sense. So this is RUNX1 versus TET2 over what the population was for RUNX1 in the bone marrow. So in other words, if you have a number above 1, above this dotted line, you had more of them than were present in the RUNX1 cells proportionately within their own population by absolute number. So when we look over the myeloid lineage as we leave hematopoietic stem cells, we see a marked increase to up to 10-fold what RUNX was, 20-fold what RUNX was, massive expansions. When we look at lymphoid lineage, it's a little more populated because these mice had more cells in general post-competition, but it's notably depressed compared to what the myeloid lineage was. And when we look at megakaryocyte erythroid, we only see one spike, which is MPP2, and MPP2 is a population that hasn't completely biased. It's split between myeloid and megakaryocyte. It hasn't picked a differentiation identity. It leans a little MEP. So in other words, we see massive myeloid expansion, and the other populations are fairly suppressed within the RUNX1 TET2 setting compared to the RUNX1 setting alone. So having characterized that, I wanted to look at whether there was any effects from host RUNX1 cells. So as many of you are aware, the bone marrow niche is complicated as many different cell types next to these sinusoids, osteoclasts, these different other stromal populations like your CAR T cells and your CAR cells, not T, that's different, and your LEPRs, which all contribute by signaling to HSC proliferation, dominance, and in the FPD setting, all of these cells are going to be RUNX1 mutations, which is different than what would occur if we were looking at a setting without RUNX1 FPD. And so this dark purple line are mice that were transplanted into RUNX1 mutants. So they're non-hematopoietic, their stromal cells are all RUNX1 mutated. Whereas this pinkish line, lighter violet, is the same cells inside a wild type recipient. And so what we can see here is both have hematopoietic RUNX1 and RUNX1 TET2s, but if you have non-hematopoietic RUNX1, you do end up about 10% more myeloid, and that was true in both transplants I did over time. And while there's multiple subsets that were changed, one of the ones that I thought was most interesting, as some of others have shown, is classical monocytes, which are considered your pro-inflammatory subset of monocytes and macrophages. And these I found over time were specifically increased within the RUNX1 mutant setting. And so we saw this pro-inflammatory bias. We see this within the bone marrow as well. Classical monocytes / macrophages are elevated in the bone marrow if you're transplanted into a RUNX mouse inside the RUNX1 TET2s. But your anti-inflammatory Ly6E lows are not biased in the same manner.
1 minute till Q&A.
But what we didn't see was any differences in expansion. As a matter of fact, expansion is just as dominant in RUNX1 TET2, both these purples, over RUNX1 in either setting. So this suggested the mechanism was independent of the RUNX1 niche. So we did a few mechanisms to test expansion. We did BRDU injection and then followed it and found that RUNX1 TET2 cells are more proliferative than their RUNX1 counterparts. We found ex vivo that RUNX1 TET2 cells are less apoptotic than their RUNX1 alone counterparts. And we also found that if you serially plate these cells again and again and again, that RUNX1 TET2s have dominant ability to self-renew over time and keep maintaining their stem cell population. And this has led to the updated hypothesis that RUNX1 and TET2 matters predominantly from RUNX1 within the hematopoietic portion and that through a variety of these pathways is driving dominance and expansion from the top of the hematopoietic hierarchy. Remaining in this work is I have done single-cell RNA sequencing on these different mice progenitor subsets, and I'm essentially trying to target what I think is driving this expansion from that once I fully analyze this UMAP to the right. And with that, I'd love to acknowledge my lab, some of our wonderful collaborators and funding sources, and open the field to questions.
Great, thank you so much, John. So there were some questions in the chat regarding the CRI that you used, and I don't know if everyone saw the comments, but maybe you could let the group know about the CRI you used.
Yeah, happy to. So this is inside an SCL CRI. So these mice are treated with tamoxifen. Both are just to keep it controlled beforehand. And excision is partial in these mice. It is not full, and it comes from the HSCs downwards. So because of that, I also quantify TET2 mutations over time directly instead of just TD tomato, because not every TD tomato cell is floxed. But I didn't show that here because it takes extra time
Yeah.
to show.
Do you know when you say you quantify mutations, are you able to actually tell whether you have a heterozygous, so one flox allele still?
Yeah, great question. The way I'm quantifying is by qPCR. So it is not able to tell the percentage of hets versus homozygous. Presumably both are there. And from a little bit of preliminary data, this isn't as strong. I think homozygous has much more profound effects in this model than hets. Hets seem to have an effect, but the expansion is a lot faster in homozygous.
Mm-hmm. A question from Suyan regarding whole bone marrow transplant or sorted cells.
In this case, this is a whole bone marrow transplant.
Great. Any other questions from the group here?
Sorry, I didn't use the reaction button.
Oh,
can I jump in?
go ahead. Yeah, please jump in.
Thanks. John, this is great work. I had a kind of an SCL question, and I think it might be similar to what Suan's asking. How much excision do you think you're getting prior to transplant? And do you think there's a chance you're going to have tomato negative cells that are only RUNX in that transplant that conflate things? Or are you purifying the tomato positives in a way that we're not going to get conflation from the other arm that's tomato negative?
Yeah, that's a great question. So 2 notes on it. I find that the excision of TET2 is very low in frequency compared to the excision of tomato. So I can give a dose of tamoxifen that makes it 95% tomato positive and 5% TET2 excision. So tomato excises much more easily than TET2 does. We target about 50% excision starting. That seems to be what the mice can tolerate in terms of tamoxifen. But I've also done this study where the second study, I don't show it because it's easier to say it without it, is actually the CRI is driving tomato expression on a VAV CRI in the RUNX1 setting for this one. So all RUNX1 cells are labeled and all RUNX1 TET2 cells are unstained. And so this one has
so your 1 to
no
3 is with a
possible
constitutive
background.
VAV-GRI. Got it. Okay.
Yeah.
Cool.
Yeah,
Super.
this
Thank you.
one's- Yeah.
Yeah. That's
Thanks, Robert.
Bobby, Lucio, you have a question. Please go ahead.
Yeah.
Just clarification for Bobby point. We're CRI driving TD tomato, not
Yeah.
TET2.
Yeah, TET2 excision is always on the SCL. Tomato in the
I
RUNX1
understand what you're talking
setting.
about.
So
you remove
every
the
cell.
tomato in that experiment from the TET2 arm.
Yes, and then
Got it.
put it onto the RUNX1 arm in the second
Thank you.
experiment,
Appreciate it.
driven by VAV.
John, Lucia here, and very nice data. And you mentioned very briefly, but I would like to expand a little bit. Have you tested or analyzed the same experiments by using heterozygous TET instead of homozygous TET deletion. So what would the results be in there?
Yeah, so we have performed some transplants in the RUNX1 setting as well. And a lot of our initial competition data was actually from the het setting. So I do see differences in proliferation, apoptosis, self-renewal. Those all exist in the het setting. That's actually what these data are from initially, but the homozygous setting has much stronger self-renewal, and it also has much better expansion over time. We're still doing kind of the initial studies of the rate at which maybe het versus homozygous expands, but it seems they have similar mechanisms at play, but they are much more exaggerated in the double excision model, from what I
If I
see.
can follow up this the question, and the TET2 flux mice without the RUNX1 mutation, eventually they develop some phenotypes, and has been published by Omar and some other people. And are you interested in testing whether the addition of the RUNX1 mutation will accelerate that?
I am interested in whether it would accelerate that for sure.
So you're going to follow on time. So I think that it takes a few months before getting the TET2 phenotypes alone.
Yeah, we have a parallel project that I've started, which is on the SCL tamoxifen induced just alone at a certain percentage, and we're following those mice in time to see how the percentage increases without being in the setting of a transplant, and whether that drives expansion. And I've also sacrificed some of those mice, and they do have higher rates of hematopoietic stem cells within their hets for RUNX1 TET2s once they get old enough. So there does seem to be some expansion, but we're definitely interested in following whether we can have a non-transplant model of heterozygous expansion and how it differs from TET2. Though weirdly, some of the things that we canonically see in TET2, at least in hets, become less pronounced outside of the transplantation setting. So that can also occur, like some aspects of how they myeloid bias, for example, like some neutrophil production. But that's still preliminary, and I'm still interpreting and gathering more data on that.
John, I'm sorry to cut you off now, but I think we need to move on. Thank you. Great talk. Thank you for sharing the latest from your work. And we're going to move on, to another Anupriya Agarwal Lab member, Luiza Ostrowski. Please go ahead. We can't hear you. I don't know if you're on mute.
Hi, can you hear me now?
Yes, we can. But we lost your slides.
Yes, I'll share that again. Yes. Can you see? Okay. Yes, hi everyone. Today I'm going to present some of the work we have been developing at the Agarwal Lab at the Knight Cancer Institute at OHSU. And as John briefly mentioned, the emergence of certain secondary mutations in the hematopoietic stem cells can cause the clonal expansion associated with clonal hematopoiesis. And the RUNX1 carriers without hematological malignancies have an increase in the prevalence of clonal hematopoiesis at all ages compared with control populations. With that in mind, the goal of our study is to understand the mechanisms driving the clonal expansion, considering the age-induced inflammation of RUNX1 FBD individuals. For that, we plan to characterize the cell populations in the bone marrow of healthy and FBD individuals, then identify the changes in the transcriptomic signature related to inflammation and immune modulation, and lastly, investigate the patterns of clonal evolution in RUNX1 FBD. Since we are interested in understanding what is happening in each cell population individually, we decided to use different single-cell methods so we could characterize the cell populations involved in the RUNX1 FPD phenotype. So we received bone marrow samples from collaborators for 21 FPD and 5 healthy patients, for which we sequenced for site-seq, DAB-seq, which includes the joining profiling of DNA and cell surface phenotypes, and single-cell attack-seq to understand the changes in the chromatin pattern. The wet lab part of the work was mostly done by Aishwarya from our lab. And since we had an imbalance in the number of FPD and healthy samples, we also decided to integrate our data with publicly available healthy bone marrow samples. So for our first goal, we integrated all of our samples, and after quality control and doublet removal, we annotated the cell types based on the gene expression of marker genes and other available data sets. And when we look at the proportion of cell types per condition, we observed a significant decrease in the TCD4 naive and TCD4 memory, as well as dendritic cells and erythroids in the FPD samples in comparison to healthy. For the CD8 memory T cells and the classical monocytes, we observed an increase in the FPD samples, which is consistent with previously published data from our lab. And although not significant, the FPD samples also presented a slightly increase in the GMP cluster. Since the clustering in the UMAP can be arbitrary, we decided to perform a differential abundance analysis where we tested for significant changes in the cell abundance across conditions. So in this graph, we can see the nodes or dots represent a neighborhood, and each line shows the cells that are shared between neighborhoods. And the pink color shows the neighborhoods that have a differential abundance in the FPD samples, meaning that they presented more FPD than healthy. FPD neighbors are concentrated in the myeloid clusters and lymphoid clusters. When we look at the differential expression by cell type, the neighborhoods colored in pink now represent the significant increase in the FPD samples, as we can see for the classical monocytes and TCD8 memory compartments. Moving on to our next objective, now we plan to look at age-mediated changes in the RUNX1 FPD samples. For that, we separated our samples into young, adult, and elderly. And when we look at the proportions of cell types for the age groups, we can see an increase in the classical monocytes in adults and elderly, as well as decline in the number of progenitors and NK cells. When we plotted the proportion of each cell population per patient, we can see that for the progenitors compartments, including HSCs, GMPs, and CLPs, there is a progressive decline in this population over aging when compared to healthy, even though young and adult presented with an increase with a higher proportion. When we decided to look at the gene set enrichment analysis for the HSCs across the age groups, we saw that when we compared adult and young, we can see there are several inflammatory pathways that are upregulated in the adults, including TNF-alpha, PI3K, and mTOR, while the young individuals presented with an upregulation in the interferon pathways. When we compared the elderly with the adults, we saw that the elderly individuals had an increase in proliferation and cell cycle-related pathways. When we look at the proportion of the myeloid clusters per patient, we can see a significant decline in the classical monocytes over aging in the healthy samples, but not in FPD, which presented an increasing trend. For the non-classical monocytes, there is a positive in the controls, which also doesn't happen in the FPD samples, where the proportion stays constant across age. Then we decided to look at the lymphoid clusters, where we saw a significant increase in the proportion of CD4 naive and CD4 memory in the healthy samples, which was also not followed by the FPD samples. And interesting, for the NK cells, we can see a decline in the proportion of the cells overaging in FPD, suggesting an impairment of the immune system in RUNX1 FPD. And to better understand this immune system modulation, we decided to run an enrichment analysis focusing on immune system gene sets. So interestingly, we observed an upregulation in almost all the pathways we tested in the NK cells of adults in comparison to young and elderly, including immune activation and cell differentiation. Now for the CH-mediated changes, we used the secondary mutations of the patients to determine if they have hematopoiesis or not, considering a VAF threshold of 2%. And when we look at the cell proportions, the cell samples that are, CH positive presented with an increase in the differentiated cells, especially including the myeloid and lymphoid compartments, and a decrease in the proportion of the progenitor populations. When we performed gene set enrichment analysis in the classical monocytes cluster, the CH samples had an upregulation of several pathways related to inflammation, including STAT and TNF signaling, which is expecting, knowing that the classical monocytes have a pro-inflammatory role. So lastly, our final objective plans to look at the single-cell DNA data to identify the patterns of clonal evolution. For that, we plotted all of our patients by age and included the mutations they presented, both somatic and germline. And we can see that the older patients presented with more mutations. When we look at the clonal phylogeny for these patients, for two of those patients, those older patients also presented with a more complicated pattern of clonal evolution, which with an increased number of clones. So based on our results, we can conclude that the RUNX1 FPD adults have an increase in the inflammatory response in comparison to young and elderly, specifically the one mediated by PI3K, mTOR, and TNF-alpha. And the changes we observed in the proportion of the myeloid and lymphoid compartments overaging can provide us with insights into the immune mechanisms driving the RUNX1 FPD phenotype. And the analysis of the CH positive individuals suggested the involvement of the classical monocytes in the inflammatory response associated with cases. As for future directions, we plan to perform cluster-specific analysis that can help us understand the clonal evolution during aging, especially considering the prevalence of CH mutations, and the analysis of chromatin accessibility with our single-cell ATAC-seq data can contribute to investigating changes in that genetic signatures and help us identify targetable pathways to prevent the clonal expansion. Thank you.
Thank you so much, Luiza. We are ready for questions. Len, I see your hand up. Please go ahead.
Yeah, I was just wondering, do you think the monocytes turn into macrophages?
Yeah, that's something that we thought about it as well. It's definitely something I want to look into if we have enough data coverage to check that differentiation.
Mm-hmm.
Yeah.
Yeah. I also wondered,
sorry, Len, can you expand as to why you're asking that question?
well, we published a bunch of papers on macrophages helping
the stem cells?
self-renewal. Yeah, helping self-renewal of the stem cells. And we have a mechanism, there's an globulin, they're on the surface kind of monitoring whether to proliferate essentially or to get killed. And so I guess I was wondering if the macrophages were activated, whether they're phagocytic, whether they're responding to calreticulin. I thought it was interesting the NK response you have also could be a calreticulin response. So I think it's worth pursuing, and if some of our mechanisms would help understand the proliferation could be interesting.
Absolutely.
Yeah.
Bobby, you were next. Please go ahead.
Yeah, I was great data. I was particularly interested to see that it looked like with aging you had kind of almost a shutdown interferon alpha, interferon gamma at the expense of TNF, and I was kind of surprised to see that. I might I don't know if I should be surprised or not, but I would have kind of anticipated the interferon alpha to go up. Do you have any thoughts on that, or maybe where, like, which are there subpopulation of cells that are not experiencing that signal, or do we think that signal is decreasing with aging? That one just kind of surprised me.
Yeah, it
Um,
was a good question. I think we were also surprised when we first saw.
okay.
But yeah, I'm not sure if it is decreasing with aging or not, or if it's just changing. Maybe the inflammation is changing and is not as mediated as with TNF anymore.
Okay.
Yeah.
If I could ask just a quick question on the single cell DNA analysis, I'm just curious if you saw heterozygosity versus homozygosity, in particular for your B core mutants. I know it's maybe only one patient there, but did you see an abundance of homozygous clones versus heterozygotes in the single cell analysis?
That's very interesting. Yeah, I can check that for sure. I don't
Okay.
think we looked into B core specifically yet.
Right. Thank you.
Yeah.
Can I add something for Bobby point? So this is HSC, right? So it's still possible that expected pattern of interferon observed in different T cells or immune cells. So, and again, cytokine response by single cell transcriptomic analysis, like coverage issues, like that could be still a point, but within HSC, it would be interesting if it's validated.
Yeah, I don't think it's wrong by anything. It struck me because I'd expect the complete opposite result, which
Yeah.
that's why we talk about these things. So
Right. We'll have time for one more question, and Grover, your hand was up first, and Wenbin, maybe you could type your question in the chat. Sorry about that. Go ahead, Grover.
Mine's a quick one. The old versus young slide. You didn't tell us what the clinical state was in those particular cases? Was, for example, the old patient, did they have MDS or AML, that kind of thing?
Yeah, all the patients we have are just RUNX1 FPD. None of them had transformed as far as we know, at least at the time of the sample collection.
Right.
Yeah, it's definitely on our radar to check if any of those patients transformed on a later stage or something like that. So
Good.
we can
Thank
ask analysis.
I'm always putting myself on mute. Thank you, Luiza. Great talk. And we're going to move on. Please continue to use the chat for questions and Q&A, and we can certainly talk about it at the end of the day as well. Our next speaker is Aditya. Forgive me if I'm not saying your name right. Singhvi from University of Pennsylvania, from Bobby Bauman's lab.
Hello, can you hear me?
Yep.
Perfect. All right, let's get this started. Hello, everyone. My name is Aditya Singhvi. I am an undergraduate at the University of Maryland. I really want to thank everyone at the conference and for Bobby for giving me this opportunity to present here today. And I'm going to be presenting the work that I completed at Bobby's lab over the summer. Let's hop into some background knowledge. So, as everybody knows, a very big feature of RUNX familial platelet disorder is the increased inflammation, which also causes an increase in sorry, let me let me run that back. It's also known that RUNX1 mutant cells, specifically neutrophils, have a higher propensity for producing cytokines. And these neutrophils, due to the increased production of these bacterial product receptors, you can see here in green the TLR4, toll-like receptor 4s, and these bind LPS lipopolysaccharides, which are a product of bacterial cell walls. And oh, sorry, I can't see my mouse. In TLR4, this upregulation of it makes the cell more and more sensitive to LPS, which causes a higher output of cytokines. Oh, sorry. And not only is this inflammation subjected to toll-like receptor 4, but there are many other aspects of the loss of RUNX1 that could lead to this inflammatory response. And this could even happen in earlier stages of the cells, such as like the hematopoietic stem progenitor cell stage. I'll be calling them HSPCs from now on. That's everything we do know, but what we don't know is if cells have altered responses to these inflammatory agents, depending on the doses, their kinetics, or even like the magnitude of the responses. Those are quite unknown, and that's what we've been trying to explore this summer. Our hypothesis is that RUNX1 mutant cells exhibit increased inflammatory signaling due to heightened sensitivity and sustained transcriptional output in response to inflammatory stimuli. Now, one thing to consider is that we didn't get to testing this all the way, but I do want to show you what methods we have tried to implement and the progress we have made towards researching this hypothesis. I want you guys to imagine some kinds of responses that we thought could occur. So we considered a few different kinds of versions, some ways that this could play out. On the x-axis, we have stimulus concentration. This could be LPS. On the y-axis, we have transcriptional response, or it could be your inflammatory response. And what I'm about to show you is some potential ways that these RUNX1 mutant cells could exhibit a very increased inflammatory phenotype. So one is their sensitivity. The RUNX1 mutant cells could be more sensitive to inflammatory stimulants. We could have magnitude where at their end product is greater. We have a baseline in which the mutant cells produce more inflammatory material right from the start. And even considering that there might not be any LPS present, they're still producing more inflammatory products even without a stimulant. And lastly, we have the kinetics. So how does the time these cells are exposed to these inflammatory agents change the output? Some variables to consider here that can point us in the right direction as to why these cells are behaving this way are like the cytokine stimulation, the dose that we provide, the timing, genotype, and of course cell types. We've considered a few approaches. We have Alyssa, we have flow cytometry, single cell RNA-seq, and bulk RNA-seq. As you can see, all of these have their own downsides to them. They have their shortcomings as well. And what we need is a high throughput transcriptional readout. And let me tell you why that is. So say we want to set up an experiment. We have 4 genotypes. We want to test this, but we do want replicates. So now we have n equals 16 conditions. Of course, we want to test 2 cells as well, two different kinds of cells. I mentioned neutrophils before, and I also mentioned HSPCs. So we could be testing those 2. Now we have n equals 32. And we want to treat these cells, right? Now we have 3 treatments. A quick note, there are a lot more than 3 treatments that we could use, but we're going to stick for 3 just for the purposes of the demonstration. And these treatments could be like LPS or other inflammatory agents. Now we're up to 96, the typical 12, your typical 96 well plate amount, but we're not done yet. We still have the kinetics aspect. We have 4 different timestamps that we want to test, which now brings our condition count to 384. And lastly, the doses that we want to provide to each of these conditions. And that gives us 1,536 different conditions or samples that we could test given these right here. And it's going to be a lot more simply because there are many more variables that we could change, right? So why is this a problem, right? Like how we are now faced with a problem that we have to conduct more than 1,000 RNA isolations, which is not very ideal. It would get very out of hand. So what we did is we decided to take some inspiration from single-cell RNA sequencing approaches. First, we have reverse transcription on crude lysates using the droplet microfluidics, as shown on the bottom left, where the cells are lysed using a microfluidics device, and then you run reverse transcription on them to obtain cDNA. The second method we have is combinatorial indexing, which performs reverse transcription inside of fixed cells, and then we would lyse those cells and then obtain the cDNA through there by filtering out the nucleic acids. Right now, these are the methods that exist, but we still need to be able to sorry, we still need to be able to sample more samples in only one run, right? Like I showed you back here, we have 1,500 samples that we need to do. So what this experiment would look like is I'm going to move us now into our the procedure that we have adapted. We've adapted a drug-seq library prep. And I'm going to start off at the top left here. We start off with an array of reverse transcription oligonucleotides in a 96 well plate, where each well has a specific barcode attached to it. So in this 96 well plate, we have poly-DT primers, which are now well-specific barcodes, and they also contain unique molecular identifiers. So the benefit of this is that we have, if you have a sample in well A1, and you have a sample in well A5, those two can be distinguished. Samples in A5 and like D7 or E7, they could all be identified separately. Yeah, so our first step would be to treat these cells with different conditions or stimulations. The first option is isolating pure RNA. We want to move away from that and try to pursue the crude lysate and fixed cell side. After treating these cells with the proper conditions, we want to perform reverse transcription on them. And this would happen after they are, we would perform reverse transcription after the samples have been mixed with the primers. So I've lost my mouse, I'm sorry. After performing reverse transcription, we would end up with this cDNA, as you guys can see here. And all of this cDNA would have a well-specific barcode, which means that every single sample, all the cDNA that was produced, is now able to be identified separately, so we can pool all of this together and then amplify it. After amplification, we can then tagment this and then proceed with your typical tagmentation library preparation procedure. And then after that, we can send these samples out for sequencing. So an experiment would look something like this, right? Here's a short schematic. We have 8 plates. We can treat them with various different kinds of titrations of drugs or different time points. We can purify them and then use barcoded reverse transcription, like I just mentioned, to pool everything together, pool all the cDNA to make a library. And then when we tagment it, you'll get a second barcode, which now gives us 8 sequencing libraries, each that has 96 samples. With these 8 plates, this gives us around half of what we want out of that 1,500 that I initially brought up.
1 minute till Q&A.
Perfect. All right. So we've used Lin-negative mouse HSPCs. We've used IL-3, TPO, interferon alpha, gamma, and IL-beta. Here is some results from a TAFE station. After cDNA amplification, we really like this curve, and after this, we can tagment this genetic material. After we tagmented it, we found and sent it out for sequencing. We found that there was successful amplification of all 24 samples. We have 6,000 to 12,000 genes identified per sample, and this makes sense because we have the genes that we were looking for were successfully identified in response to the treatments that they were provided. Lastly, I have some box plots here. When these cells were treated with IL-3, we would expect that the activation of STAT5 target genes like SOX2 and PIM1 would be upregulated, and that's exactly what we see here. And moving on, when these cells were treated with interferon alpha, we see an upregulation of IRF7 along with MX1, which is a downstream marker for interferon alpha signaling. Moving on, we have this gene Ly6A, which is also known as SCA1, and there's an upregulation of this as seen when treated with interferon alpha. And lastly, we have Ly6C2. This is a differentiation marker, so this is more important to us when we're studying various time points. So when we begin working with larger time maps, where we're looking at cells exposed to these conditions for longer time periods, we can pay more attention to these. And this is more biased towards, this marker at least is more biased towards monocytes. And lastly, to finish this off, what we need to do is really look at ways to significantly optimize how we use crude lysates, and then benchmark this against fixed cells. This can really give us a good idea about which method or which approach is more cost effective, since this is the primary purpose. We're trying to establish a low pass and high throughput method. So yeah. I would like to thank everyone. I would like to thank Bobby, especially Eiley Klein, Amanda Versace, Meredith Arterburn, and Shreya Gounder for helping me throughout this summer. And I will now open the floor up to any questions.
Great, thank you, and well done for an undergraduate student. Very well done.
Thank you.
It's exciting that you've set this up. I think this will be really powerful to have a platform like this, and yeah, I'm looking forward to seeing what you are able to do with it moving forward, Bobby and team. Any questions for Aditya? All right. Thank you. Okay, well, we will move on to the next speaker, Dr. Alyssa Parker from Alex Bick's lab at Vanderbilt. Please take it away.
Hey, awesome. Thanks, Katrine. All right, does slide sharing look okay? Okay, great. All right, well, I'm excited to share with you all today a case report about an intersection of RUNX1 familial platelet disorder and familial idiopathic pulmonary fibrosis. So familial platelet disorders are something that now have been studied for about 50 years. Some of you may recognize this paper, which was the first to report on a family with what was eventually diagnosed as RUNX1 familial platelet disorder. And with the RUNX1 research program, we've had the pleasure of being able to work with this family in ongoing research. This family has been identified to have an early stop codon at the 287th amino acid in RUNX1. And we see that throughout many generations of the family, there have been several people who have had AML and MDS, and many people that are carriers of this RUNX1 variant. And so in the presentation today, I'll be focusing on this woman down here on the left side of the chart indicated by the arrow. So this patient was a woman who passed in her 60s. She had RUNX1 FPD, and so throughout her life, she had chronic thrombocytopenia, and she also had idiopathic pulmonary fibrosis that eventually was the cause of her death. On the right, you can see some stains that are showing the extensive fibrosis that we saw in her lung. So when she passed, she was really interested in research and asked if she could donate her body to research. And so we were really thrilled about the possibilities of what we could do with her samples. And part of the reason we were so excited about this is because we know that RUNX1 FPD can manifest in various tissues, which makes us think that RUNX1 variants play a role in many tissues. We see things in RUNX1 FPD that have to do with blood, as well as the skin. We see people have migraines, things like asthma and GI complications. And so really several tissues that we could investigate and see effects of this RUNX1 variant potentially. So we performed an autopsy on this patient and were able to collect samples from 11 different tissues. We sampled lymph node, lung, liver, kidney, mesenteric fat, bone marrow, heart, lung, spleen, colon, and skin. And so this specific variant that the family carries is this early stop codon here that is positioned towards the end of the gene, but in a position that we would anticipate would still result in nonsense-mediated decay, meaning that RNA transcripts carrying this variant are thought to be destroyed by the cell. And so we anticipate that in all of these different tissues, we would have this RUNX1 effect of having this nonsense-mediated decay of the variant. And so we would anticipate that we're able to see this effect in various tissues. So the question that we were really excited to ask was, how does this RUNX1 variant affect tissues throughout the body? So we performed single-cell RNA sequencing on all the available tissues that we had. We were able to collect thousands of cells from these tissues and sequence them one cell at a time. And so we were really interested in what we were able to see globally, but also in investigating the lung because that was her cause of death, and so collected many samples from the lung. And so first we just wanted to dive in, what did we see in her lung that maybe could give us some clues? And so something that we noticed really strikingly was that TGF-beta signaling seemed to be increased in her lung. Here I'm showing relative signaling strength comparing to a different patient who had IPF but did not have RUNX1, as well as samples from various regions that we sampled from her lungs and then compared to tabula sapiens, which is a healthy control. And so we saw that she had higher TGF-beta signaling than we saw in our controls. And this is something that we thought was interesting, as we know that TGF-beta is related to fibrosis, but also was kind of surprising because there have been some studies that have shown that RUNX1 overexpression seems to trigger TGF-beta signaling normally, but we anticipated that we would have lower expression of RUNX1 considering the variant in this family. So then we looked back to try to evaluate what was actually happening with RUNX1 expression. And so we saw that surprisingly, RUNX1 expression in this patient's lung was high. Here in the middle are her samples, and then we're comparing to IPF lung and the healthy control. And we saw that surprisingly, she had very high expression in all of those samples that we collected. And when we looked throughout the samples from her body, we saw similarly that RUNX1 expression was relatively high compared to controls. So this kind of made us question, is nonsense-mediated decay really happening as we think it is? And so we used bulk RNA sequencing on 4 different tissue samples to then look at which allele was being expressed. And here the mutated allele is the pink one or the one in pink that is T. And we saw that that allele was still expressed, and it was expressed almost at 50%, meaning that there really wasn't that much nonsense-mediated decay occurring. And so in thinking about how this might come about, we thought about alternative splicing, different ways that the exons can be pieced together. And if there are different alternative splicing patterns happening, that could change what really is the end of the transcript, and then could change whether nonsense-mediated decay is initiated or not. And so this is something that we're continuing to think about and are considering doing long-read RNA sequencing to capture the splicing patterns. So something else interesting that we wanted to consider is that this patient also has a family history of IPF, but now coming from the paternal side rather than the maternal side where we saw the FPD family history. So we wanted to see if there was some kind of genetic cause that we could identify for this family history of IPF. So we performed whole genome sequencing on both our patient and her parents, looking for variants that were seen in both the daughter and the father, but not in the mother. And we were really thrilled to identify a pathogenic variant in a gene called ABCA3 that has been linked to lung disease that was carried by both the proband and her father. This was a splicing variant in a gene that's known to play a critical role in the production of surfactant. And so we were really excited about this until we noticed that the inheritance pattern is autosomal recessive. And if we think about our family history, it doesn't really seem to be an autosomal recessive inheritance pattern. But interestingly, we saw in the literature that there have been some reports that when patients inherit both a variant in ABCA3 and a variant in a different gene, SFTPC, they have lung disease. And so I'm showing here some pedigrees from a study that found kind of these 2 variants coming to patients from different sides of their family. In the dark quarter, that's indicating variants in ABCA3, and then the light quarter is in SFTPC. And we see that parents who had these variants had no symptoms of lung disease, but then the children did have lung disease. So we wondered whether our patient had something similar. So we didn't see anything specifically in FTPC, but we did find a promoter variant in SFTPA2. So these are the genes that encode surfactant. This is surfactant protein. So SFTPC is one component, SFTPA is another component. And so this is a variant that we saw in the promoter of surfactant protein A2. And so we considered, or we hypothesized, that potentially inheritance of this variant in ABCA3 with SFTPA2 may induce pulmonary fibrosis. And we saw both of these in the proband and her father, which then could create that inheritance pattern that we are seeing. So we went back to our samples from the patient's lung and looked at expression of this gene, SFTPA2. And when we compared to the distribution of samples from GTEx, which is healthy controls, we saw that our patient's value, which is indicated by this red line, was very low compared to the rest of the population, indicating that it is likely that this promoter variant is having an effect on the expression. So we now plan to validate the effect of this gene, or the effect of this variant on expression of SFTPA in lung cells, to be able to follow up on the possibility for this effect. So just to conclude, in this work, we were able to investigate further this RUNX1 variant in this family, and now have this hypothesis that it may be escaping nonsense-mediated decay, potentially through alternative splicing. And we also identified variants in ABCA3 and SFTPA2 that we think in combination may induce pulmonary fibrosis. So I just would like to acknowledge the various members of this team who have worked on this project, as well as the RUNX1 Research Program and the Chan Zuckerberg Initiative that have made this work possible. So happy to take any questions while there's time.
Thank you so much, Alyssa. Great talk. Do we have any questions for Alyssa? While we wait, I'm curious, after having, I think you were there yesterday, right, Alyssa, when you heard Dr. Waterfield's talk around the impact of RUNX1 loss on MTEC cells, and just curious whether you might think, and then we heard from Paul Liu that there might be, that lung pathologies are not insignificant, or there is a prevalence in our RUNX1 cohort. Any thoughts that RUNX1 might be contributing to the lung disease in this individual as well?
Yeah, I wish we were able to tease apart these a little bit more clearly. And I feel like we've been able to find some things like this TGF-beta signaling that seems like maybe this could be somewhat linked to RUNX1. But I feel like without being able to really separate what is caused by RUNX1 versus maybe some of these other variants that are coming from the other side of the family, we can't super confidently say just with this example, like what's the cause.
Yeah.
But I think with some of these follow-up validation experiments, maybe we'll be able to get at that a little bit more clearly, hopefully.
Yeah, absolutely. All right, Nancy, you have a question?
No, just to comment that TGF-beta signaling also activates RUNX1, and together TGF-beta signaling, RUNX1 and Notch induce sort of an EMT-like processes. So, which could be, so I think that TGF-beta and the RUNX1 thing could be quite, linked functionally.
Yeah. Okay. So you're saying like if this patient had some fibrosis that was occurring because of whatever other family history, that could then induce the higher expression of RUNX1 that we're seeing?
Yeah, or it could be that the elevated TGF-beta signaling is contributing to that higher expression of RUNX1.
Interesting.
And that the two together could be contributing to the fibrosis.
Yeah. Okay. Well, thank you.
Great, thank you. Any other questions before we move on? I guess we probably should move on so that we stay on track.
Hey, Esther and I have the same question because I was about to ask,
Oh,
like,
go ahead.
do we think it's spliced to a protein product? I'm just reading Esther's question at this point, but the splicing leading to aberrant protein production or a different protein product, or any thoughts or hopes or guesses?
Yeah, so not sure about the proteins yet, but at least what we're seeing from the splicing patterns just from our bulk RNA, it does seem like there are transcripts that include like the very last exon and there are transcripts that don't. And so potentially that could result in different proteins, but we'll have to follow up on that further.
Super interesting. Thank you.
Okay, thank you.
All right, and our last speaker of this session is Dr. Sawsan Sulaiman, who's coming to us from Children's Health Ireland. Welcome.
Thank you so much. I'm going to be taking you a little bit away from the heavy scientific part, and it's just an interesting case that I came across as a pediatric registrar working in Ireland. So it's the next slide. So I'm going to be presenting a patients with neurofibromatosis type 1, Moya-Moya syndrome, RUNX1 familial platelet disorder with predisposition to myeloid malignancy. Next slide. So as you're well aware, there is two types of neurofibromatosis, type 1 and type 2. And in this presentation, we're going to be concerned with type 1, which is an autosomal dominant disorder caused by mutation in NF gene in chromosome 17. It characterized by multiple manifestations affecting various parts of the body, including the skin, ocular, skeletal, and neurological manifestations. There is multiple complications to neurofibromatosis type 1, and cerebral vasculopathy is one of them, and it's becoming increasingly more recognized now. However, Moya-Moya syndrome in particular is exceptionally rare at 0.6%. There's Moya-Moya disease and there's the Moya-Moya syndrome. The Moya-Moya disease is when there's a progressive occlusion of one of the cerebral vasculature. It's called syndrome when it's part of or as an association of an underlying cause, such as neurofibromatosis in this case. Yep, next slide. Next slide, sorry. Yes, so RUNX familial platelet disorder with association to myeloid malignancy, as you know, causes prolonged bleeding. The patients sometimes present with easy bruising, and it increases the risk of hematologic malignancy. It's also characterized by thrombocytopenia. The platelet size is usually normal. The bleeding is usually greater than expected because it's a qualitative platelet dysfunction. Next slide. So in this case, we're going to be discussing the presentation of the patient, the diagnostic evaluation that was done and how we managed it. And we're just going to illustrate the complexity of balancing between a bleeding risk, such as in this case, and versus the ischemic stroke risk. And we're going to emphasize a little bit on the importance of a highly individualized multidisciplinary approach. Next slide. So moving on to our case, Ellen is a 17-year-old Irish female. When she presented, she was 2 years of age. She presented to the local pediatric team due to developmental delay in her milestones, and she had profound speech delay. Other than that, she had normal clinical background. She had mild childhood asthma that was well controlled. In her family history, there is neurofibromatosis type 1 affecting both the mother and the maternal grandmother. When the physical examination was carried out, she was found to have multiple cafe au laits and inguinal frickling. All the above warranted further investigation, and genetic workup was done, and she was found to have NF1 familial nonsense mutation. Next slide. So over the course of the years, she had multiple FBCs that were done, and it was a completely incidental finding that she had persistent thrombocytopenia. Her platelets usually falls between 80 and 150, which is below the normal range. She also complained of easy bruising. So by the age of 6, she was referred to the hematology team. There's no massive bleeding or anything of that sort. And the question to the hematology team, whether that was something acquired or congenital. Moving on to the next slide. So when she met the hematology team, as you can see in this slide, she had multiple blood tests that were done. All of them were normal, and they had a genetic consult done as well as part of their workup. And their question to the genetic team was if there is absolutely any association between neurofibromatosis type 1 and thrombocytopenia. And their answer was basically no. So the management plan at that point was to watch and wait and just follow her up with annual FBCs. Yeah, next slide. By the age of 12, she hit her menarche, and LM described her periods always as flooding. They're quite heavy with clots, and she had to change her sanitary pad once every hour in the first 2 days. That created many days of school absence, and it impacted her life significantly. Subsequently, she developed iron deficiency anemia, and she was treated with oral iron supplements. Next slide. She was immediately linked with the gynecology, who tried 2 medications. It was a tranexamic acid, which is an antifibrinolytic, and she was tried also on combined oral contraceptive pills. She tried each medication separately. They put them as a combination with breaks. all in all, it gave her minimal improvement. At the time, because she was only 12, she was reluctant to having an IUD. It's a little bit uncomfortable procedure for a young girl. So she was reluctant at the beginning. Next slide. It's worth mentioning that her mother, who we mentioned earlier, has background of neurofibromatosis as one as well. She was diagnosed with poor prognostic acute myeloid leukemia, and she was she underwent allogeneic bone marrow transplant in a different institution. As you know, neurofibromatosis type 1 is known to have multiple neoplasms as part of the complications, but hematological neoplasms is not very common. However, leukemia and lymphoma were described in some of the patients. Moving on to the next slide. By the age of 15, she started complaining of severe headaches that were consistent with the description of migraines. So she was treated as such, but the headaches were still persistent despite the medications. And an MRI was done on April 2022, and she was found to have Moye-Moye syndrome, which, as you can see on this report, it's a significant hypoplasia of the left internal carotid artery and the left middle cerebral artery. So she was linked in immediately with the neurology team, and they advised her to start aspirin 75 milligrams daily to reduce the ischemic stroke risk. So from this, you can see that we have a treatment dilemma now. Next slide, please. The question is whether to treat her menorrhagia by continuing giving her the combined oral contraceptive pills, but potentially increase the risk of thrombotic of ischemic stroke, or to start the aspirin, reduce the ischemic stroke, but exacerbate her menorrhagia. Next slide. So therefore, multiple MDTs were carried out to manage this case, and they were all wrapped around managing her heavy menstrual bleeding while balancing the increased stroke risk. And like we mentioned earlier, she was having a poor quality of life because of the menorrhagia. So it's really impacting her social life, her school attendance. And she was emphasizing on that a lot, the patient herself. So eventually, after multiple meetings with the patient and her parents, it was the conclusion that the risk of ischemic stroke outweighed the bleeding risk. So aspirin was initiated. Luckily, she had successful neurofibromas excisions done, and there was no bleeding. And at the time, she was still on aspirin. Moving on. So a person might ask why not to try alternative medications, which they were considered, like progesterone-only pills. You will avoid estrogen, which is great. You're reducing the thrombotic risk. However, there is a limitation to the fact that it's used daily for a teenager. That's kind of hard. And it does cause intraministerial bleed, which again, for a teenager, is not a great look. Tranexamic acid was the limitation here, that it was used before, and also the studies have shown that it's not quite effective with heavy menstrual bleed. Intrauterine devices such as Mirena coil is the best option for menorrhagia, but due to her young age, she was reluctant initially, but now she opted for one now that she's 17 years of age. Next slide. So by the age of 16, she was diagnosed with the pathogenic RUNX1 familial platelet disorder. It was found, and she was also found to have persistent monocytosis on her FBCs that led the hematology team to think maybe she was developing leukemia. So a bone marrow aspirate and biopsy were carried out, and they were normal. The flow cytometry was also normal. Bone marrow cytogenetics showed NF1 and RUNX1 mutations. Next slide. So the care plan was basically the 3 pillars, which is multidisciplinary approach. It's a collaborative work between the pediatricians, the hematologist, neurologist, and the gynecologist. The individualized care, which basically tailoring the medications according to her needs, preferences, lifestyle, and also to do genetic counseling, because for her to understand her condition and the implication of RUNX1 familial platelet disorder. The third one is basically monitoring and follow-up monitoring for early signs of stroke, and basically also to monitor her menorrhagia and follow up on the efficacy and effectiveness of the medication, and if there is any side effects, and just regular follow-up for RUNX1 family platelet disorder from that point of view. In conclusion, next slide, sorry. Yeah, so Ellen is now 17 years of age, and she had a background of neurofibromatosis 1 and Moye-Moye syndrome. She was later diagnosed with RUNX1 familial platelet disorder, which explained her persistent thrombocytopenia. And we presented this case to you today because it's a complex medical scenario where you have two rare conditions in one patient, and it created a therapeutic dilemma. And it exemplifies exactly the highly individualized multidisciplinary approach to balance between the two compelling risks of stroke and bleeding. I would like to thank so much my supervisor, Dr. Beatrice Nolan, who was supposed to attend, but I think she's busy. Thank you so much for your time, and I'm happy to take any questions.
Thank you so much, Dr. Sulaiman. That was great and a complex case indeed. I have the same question it looks like that Esther has, which is whether the mother, given she had the hematologic malignancy, whether she was tested for RUNX1 FPD, or did the RUNX1 FPD come from the paternal side, or is she de novo?
I think she is de novo. I don't think she has RUNX. That's from what I understood from my hematology consultant. I don't think the mom has RUNX, so it's not related. Yeah,
And
it's just
any idea whether the father might, or unclear?
No, I'm not quite sure, but I know that the family has underwent testing for after she was diagnosed with RUNX1.
Got it. Got it. Any other questions from the group? It's good to hear that sounds like she's being well managed and that there's a whole multidisciplinary group on top of things.
Exactly. And now she's transitioning to the adult care, but she will be still followed up from RUNX1 point of view with the hematology, pediatric hematology still.
Great. All right. Thank you. Thank you so much to our speakers this last session. And also a nice variety of different talks. And it's great to see all the ongoing research. So we now have a break. We actually have an extended break because Dr. Tim Ripperger unfortunately has a family medical emergency. So he will not be presenting as the first speaker in the next session. So our break will actually go from now until 10:50 a.m. Pacific, which is 1:50 p.m. I think I got that right. 1:50 p.m. Eastern. And then if you're anywhere else in a time zone, I'm going to ask you to calculate that. All right, so thanks for everyone for being here, and looking forward to the next session, which we'll have at 10:50, Dr. Kat Menezes from the NHGRI will be kicking us off. Until then, welcome back, everyone. And we're going to kick off the next session with Dr. Ana Catarina Menezes from NHGRI. And she'll be presenting and talking to us about multi-omics analyses, which reveal transcriptomic and proteomic dysfunctions in RUNX1 FPD platelets. So welcome, and happy you could be here. Take it away, Dr. Menezes.
Hi, everyone. Thank you for the invitation to be here again. I'm really happy to be able to show our current data on RUNX1 germline variants and how we are applying multi-omics approaches to study RUNX1 FPD platelets and how that can help us with RUNX1 variant classification. So, the next slide, yeah. So I don't need to give too many details here about what RUNX1 FPD is to this audience, but I would like to emphasize its characteristic features, which include mild to moderate thrombocytopenia, functional platelet effects, which will be the focus of my talk today, and markedly increased lifetime risk of MDS and AML. And given the clinical variability and the clear risk of leukemic progression in these families, it became evident that we needed a systematic effort to follow patients over time. Next slide, please. And that is why our group established the NIH Naturalist3 study on RUNX1 FPD, which provides a framework not only to characterize the clinical course, but also to collect samples that enable mechanistic studies, such as the platelet multiomics approach, which I will show you today. And I'm not going to go in more details, as Paul will talk after me. Next, please. Oh, sorry, these are some animations. About this slide, clinical and experimentally, we observed a spectrum of platelet abnormalities in RUNX1 FPD patients, most notably thrombocytopenia and impaired platelet aggregation. Here in this slide, and if you could just continue so we can see all the plots, sorry. Thank you. So in this slide, I've summarized the key platelet abnormalities in these individuals as reported in our clinical paper. As expected, platelet counts are significantly lower in patients compared to healthy controls. Also, nearly half of the patients tested here had reduced dense granules per platelet, which indicates storage pool deficiencies. In terms of aggregometry, as you can see in the lower plots, all patients showed impaired aggregation to at least one agonist. ATP release was also abnormal in all patients, particularly in response to low-dose ATP. Finally, as you can see on the right, bone marrow histology showed some degree of megakaryocytic defects in the majority of the samples, with small hypolobated megakaryocytes and nuclear abnormalities. Next, please. And here are some of the most common techniques that researchers use to study platelets. Aggregometry gives us functional responses to agonists, like I just mentioned. Then we have flow cytometry and imaging, which provide insights into receptor expression and activation, for example. Proteomics and transcriptomics let us take a global look at the molecular regulation, and electron microscopy, which allows us to see platelet structure and granules in detail. Together, these methods provide complementary perspectives on platelet biology. Some here are already done by our clinical team every time a patient comes in. So, for example, aggregometry and flow cytometry and electron microscopy. And today I'm going to focus on transcriptomics and proteomics of platelets. Next, please. So given the clear clinical evidence of both quantitative and qualitative platelet defects in RUNX1 FPD patients, there is a critical need to better understand the molecular mechanism driving these phenotypes. And to address this, we perform a multiomics analysis of platelets from RUNX1 FPD patients. And our objectives, as it's written here, were first to dissect the underlying mechanisms of that contribute to platelet dysfunctions in these patients. Second, to identify candidate biomarkers or pathways that could be targeted therapeutically. And third, to generate functional evidence that can support variant interpretation beyond the current gold standard in vitro assays. So to accomplish this, we use purified platelets from patients and healthy controls. And this is the workflow where includes bulk RNA sequencing and TMT-based quantitative proteomics of purified platelets. Next, please. So here in this slide, you can see the different platelet samples that I've used. And I've used samples from patients carrying a range of RUNX1 variants, classified as variants of unknown significance, or VUS, likely pathogenic or pathogenic. And as you can see here in the lollipop plot, is easier to see where they are located in the RUNX1 protein. In this table, we have a summary showing the classification, variant type. We have missense, nonsense, framesheet deletions, and also whether the patient has evidence of clonal hematopoiesis or CHIP or not at the time of sampling. So this in total, I've used 51 samples. So we have 11 controls and 40 RUNX1 FPD patient samples. Next, please. So first, let's start with the bulk RNA-seq data from platelets. Here we see a PCA plot, including all controls and all RUNX1 FPD samples, regardless of variant classification. And what stands out is that is the clear separation between controls and patients with pathogenic or likely pathogenic variants, staying in one side, and we have the controls in the other side. And then we have the VUS group as expected, which shows more heterogeneity, with some samples clustering closely with controls, others clustering with the pathogenic group, and some lying in between them. Importantly, these transcriptomic patterns were consistent with the clinical phenotype of the patients carrying these VUSs. And this suggests that platelet transcriptomics could serve as a valuable tool in the clinic to help interpret these patients with VUSs. Next, please. In these PCA plots, I basically remove the VUS samples since their heterogeneity can skew the downstream analysis. And again, here it's even more evident. We see that clear separation between the platelet transcriptomes of RUNX1 FPD patients with likely pathogenic or pathogenic variants and healthy controls, highlighting consistent and substantial transcriptional differences. And this separation supports the concept that RUNX1 germline variants have a broad and measurable impact on platelet gene expression, even in patients who have not developed malignancy. Next, please. So next, we explore the differential expressed genes. The volcano plot highlights over 2,200 differential expressed genes with 40% downregulated and 60% upregulated in RUNX1 FPD platelets compared to healthy controls. And when we bring in the heat map, you can appreciate the broad transcriptomic dysregulation in the RUNX1 FPD group. Interestingly, we also see emerging subclusters among our patients, suggesting a transcriptional heterogeneity. We're currently investigating whether these patterns correlate with clinical features like variant location, platelet counts, or malignancy progression. Next, please. So to gain functional insight into the transcriptional changes, we perform GCA on the differentially expressed genes. And interestingly, genes upregulated in FPD platelets are enriched in pathways related to ribosome biogenesis and early cell fate programs, including ribosomal RNA, metabolic process, cell fate commitment, etc. This could suggest a shift towards transcriptional programs more commonly associated with immature or progenitor-like states. And in contrast, downregulated genes are strongly associated with platelet-specific functions, which include coagulation, wound healing, platelet morphogenesis, etc. Next, please. Zooming in on the upregulated gene sets, we see enrichment in ribosome biogenesis and cell fate commitment pathways. This is consistent with known roles of RUNX1 in regulating ribosomal gene expression and may reflect a compensatory or dysregulated biosynthetic response. We also see upregulation of transcription factors and developmental regulators like HEF1, DKK1, GATA3, and members of the SOX and Wnt signaling families. These changes suggest a shift in megakaryocyte differentiation programs, possibly indicating a block in maturation or inappropriate activation of the early progenitor pathways in RUNX1 FPD platelets. Next, please. So focusing now on the downregulated gene sets, we observe a significant decreases in key coagulation factors and platelet receptors, including, for example, factor 8, factor 13, VWF, GP6, and ETGB3. This pattern reflects impaired platelet adhesion, aggregation, and clot stabilization in RUNX1 FPD patients. Additionally, several cytoskeletal and signaling proteins, such as MIL9, FLNA, etc., are also downregulated. These proteins are critical for platelet shape change and activation, so their reduced expression likely further compromises platelet hemostatic function. Next, please. So now we're moving on to the proteomic studies. So here we have the PCA plot of our platelet proteomic data sets, including all patients and controls, and these were using the same samples as the bulk RNA-seq. And similar to those previous results I showed, we see a strong separation between controls and patients with pathogenic or likely pathogenic variants. And again, we have here the VUS samples showing heterogeneity, some clustering with controls and others with pathogenic. And I find it quite nice because it's again is associated with patients' clinical phenotype. So this pattern mirrors what we saw at the transcriptome level and suggests that proteomics could also provide this valuable information for variant interpretation and reclassification. Next, please. And here in this PCA plot, I again remove the VUS samples, and similar to what we observed with the transcriptome, there is that clear separation between healthy controls and pathogenic variants, confirming that the proteomic profiles are distinctly altered in FPD platelets. This reinforces that RUNX1 germline variants drive broad molecular changes, affecting both RNA expression and the protein content of platelets. Next, please. And here we have the volcano plot displaying the differential expressed proteins in RUNX1 FPD platelets compared to controls. We managed to identify around 425 proteins that were significantly dysregulated with a near even split. So we have 51% downregulated and 49% being upregulated, confirming a broad and balanced proteomic alteration in RUNX1 FPD platelets, which is consistent with the extensive transcriptomic changes we observe. Next, please.
1 minute.
Okay, thank you. I'm almost done. The enrichment analysis of differentially expressed proteins highlights key platelet-related biological processes affected in RUNX1 FPD. So these include adhesion, integrin signaling, coagulation, pathways critical for platelet function. Next, please. So integrating transcriptomic and proteomic data reveals this strong concordance in dysregulated platelet pathways in RUNX1 FPD. We have key platelet functions such as adhesion, activation, and coagulation are consistently downregulated at both RNA and protein levels. We also see reductions in cytoskeleton and vesicle traffic components, which likely contribute to impaired platelet shape change and secretion. And the coordinated downregulation of coagulation-related factors supports observed defects in clot formation and hemostasis. So overall, the proteomic results validate and refine the transcriptomic findings, emphasizing the protein-level changes that are critical to platelet dysfunctions in RUNX1 FPD. Next, please. So I have here just to summarize this table where we see a multi-level disruption of hemostatic processes in RUNX1 FPD platelets, from platelet production to clot stabilization and wound healing. I'm not going to go through everything, but you can read here. Overall, these alterations provide a comprehensive picture of platelet dysfunction driven by RUNX1 variants. Next, please. And finally, to summarize, this study represents the largest multi-omics profiling of RUNX1 FPD platelets to date, both the platelet transcriptome and proteome of the selected RUNX1 FPD patients were strongly dysregulated compared to healthy controls. And I've mentioned all these different pathways related to platelet function that were dysregulated in our patients' platelets. And finally, platelet multiomics could support the current gold standard assays for RUNX1 variant interpretation. Next, please. And this is just my acknowledgements. I would like to thank everyone in my group and at NIH for all the help in this study, but most importantly to all the patients and families that made this study possible. And thank you for your attention.
Great. Thank you so much, Kat. Beautiful work. I have a question, and then Nancy also has a really good question. My question was, when you were showing the transcriptomic signatures and you had a group of FPD patients that sort of all lined up, and then you had this other group that was off to the left that looked slightly different. I don't know if there was a difference in the downregulated genes or what, but I was curious, were you able to look at that group that was different and see if they had a different protein expression as well? Like, or what was the difference between those two groups in that RNA slide that you showed?
Are you saying, sorry, in the different subclusters of patients we have in the...
Yeah, I think it was the heat map slide.
Yeah.
A cluster of FPD that didn't quite fit with the rest of the group.
Yeah. Yeah, I find that really interesting. So we have that top part of the heat map being that show red in controls and then blue in the others, being all the platelet related genes. And I find interesting our patients, separated. I'm currently trying to correlate what is the similarity between these clusters of patients. For example, the cluster just next to controls on the right, so the one that has the top differentially expressed genes quite downregulated compared to controls. Those are, for example, patients that their commonality is truncated proteins. We have their R201s, R320. So I'm currently trying to, I don't think I saw really in terms of chip or no chip. I think it was more, the variant itself. But I'm trying to connect that, because I find it interesting.
And whether you see a difference in their protein expression, right? When you look at the proteomic data.
Exactly, because that left group seems more closer to controls, and then the other. So I wonder if, what we can get from here. In the end, I would like to, can we identify any biomarkers or anything? Because doing bulk RNA-seq and proteomics in platelets is quite time consuming, expensive. So could we just find those genes that we could then test, and that would be the goal.
And great, thank you. And the question from Nancy, and it looks like Esther wanted to tag on top of it. So Nancy's question, and I think we're all very curious, there were those VUSs that sat squarely in your likely pathogenic transcriptomic profile. Also in the proteomic profile look like. Which VUSs are those? Would you be willing to share those?
Yeah, so could you go back to any of the plots?
Catherine, you'll have to go.
Please, So I think if we go, I think the proteomics actually, I love that PCA plot, is just beautiful. Those, which is concordant with the transcriptomics. So those three that are... the three VUSs that really cluster with the likely pathogenic or pathogenic are it's a missense, which is the G170R, which Kaya shown in the genomic paper that leads to abnormal splicing. But currently, I think he's still a VUS. I think he was being reclassified to LP maybe. And then the other one is a splice site variant. That is still a VUS.
Oh, that's still a VUS. Okay.
Yeah.
Oh, and Natalie's jumping in and said should be likely pathogenic.
Yeah.
Oh, yeah, it'd be really interesting to see if we could reclassify these.
Yeah.
So Esther, I think that was essentially what you're saying, and whether looking at RUNX1 expression and then in combination with the clinical picture, basically the whole sort of holistic picture, whether that's enough information and data to really push some of these into a classification, a new classification.
Yeah.
Kennedy, please go ahead. I see your hand up.
Yeah. Very nice talk. Enjoyed listening
Thank you.
to the findings that you have. We've had an interest in this transcriptome and genes regulated by RUNX1. And, about 20 years ago, we reported the expression profiling and showed many of the same genes that you found also to be decreased. So my question is, are you also going to have data on the megakaryocyte? I'm particularly curious to see if the changes seen at the megakaryocyte level are different than that we see in platelets. Are there genes that are downregulated in the platelet transcriptome, but not in the megakaryocyte? Would you be having that data also coming down the pike?
Yeah, thank you for your question. That would be lovely. I feel just with megakaryocytes is so difficult to isolate them from our bone marrow samples. The percentage is so low. So we are trying in the lab to set that up, but it's a bit challenging. But that would be really nice to see upstream changes. So we try to do that at the CD34 level. Erika, for example, in our lab, those studies a lot, but megakaryocytes, we still have to set that up. Yeah.
Thank you.
Thank you.
Katrine, you're muted.
Oh, sorry. Go ahead, Alan, as long as it's quick. Otherwise, we should probably move on.
Yeah, hopefully this is pretty quick. This is really nice. I was thinking more about clinical trials and biomarkers. How practical do you think this would be to use as biomarkers for clinical trials looking to obviously enhance RUNX activity? Doing it at this level and collected remotely, it just seems like a really good biomarker.
What would be a good biomarker? Or
looking at these the transcriptome and proteomic changes. Could you do it?
So I'm trying to find anything anything that it's easy to then confirm, for example, with just flow cytometry or something like that we can easily access. I have a few interesting targets to I think that's going to be my next step, is just exploring these key platelet genes that are being dysregulated. But something that we could potentially use like flow cytometry and quickly assess those levels. That'll be great, I think.
Yeah, that'd be really great, and could be implemented in the imatinib study, for example.
Sure.
Yeah.
To see response. I think that's what you're getting at. Yeah. Great.
Yeah, I was just wondering about doing it at this level. Good, thanks.
Thank you.
Thank you very much, Kat. Okay, we're moving on to Paul Liu and the team.
Thanks for the invitation, Katrine. You asked me to update our natural history study. So we're going to have 3 short topics. And so I'll present the first one, and then David, the second one, and BC, the last one. The next slide, please. So yeah, I will give a brief update on somatic mutations, and David will talk about hematologic malignancies in children, and BC will talk about telomere. So next slide, please. Yeah, so you have saw this slide already. You can move on. Next slide. And so this is the current updated list of somatic mutations found in our study participants. And again, sorry for the small letters, because it's a long list of mutations in these patients, and anything more than one is listed here. And B core remains the most frequent. mutated gene, and I can't even read myself. So 25 in patients with without malignancy, and 6 with malignancy have wrong B core mutations. And the second place is TAT2 and DMT3A and PH6. Interestingly, we now saw mutations in RUNX1, so many mutations. This is different from our publication. So we have seen 3 patients with RUNX1. So next slide. Show you some of the characteristics of these 3 patients. The first one have a large deletion as germline and also have MDS. And this patient was supposed to have a large deletion at the somatic mutation by an outside testing agency. And we're not quite sure if this is real. So we're still investigating whether this is the same germline or is really truly a somatic deletion, an independent one. The second patient has a substitution at the germline and had AML, and this patient also have another substitution clearly independent as a somatic at 18% variant allele frequency. And so the last patient have arginine 201 stop, again AML, and with another kind of a small nucleotide substitution leading to a frame shift. So clearly the RUNX1 somatic variants do present in our patients, but only in patients develop malignancy. And so it's not as frequent as some of the other ones like B-core and TAT2. Next slide. So now move on to David for the next topic.
Yes, thank you, Paul. That's actually a great segue into this topic. This is some work that recently came out and is actually under revision, and then hopefully a second review for publication. And work that was done by Amra Kajik in our lab, a former postbac who's now moved on to greener pastures. But one of the questions that's come up from lots of patients and their families when they come to see us is the statement that along the lines of, do we need to test our son or our daughter? Do we need to do bone marrow biopsies and things like that? Because our doctor told us that, well, the likelihood is they're not going to get cancer until they're about 35, so we really don't need to bother them right now. And what's the benefit from that? But the counterpoint to that was that sadly, and as is the case for a lot of natural history studies that have been going on long enough, we are starting to see more and more children who develop leukemias and other heme malignancies. And they're oftentimes, not entirely, but oftentimes they are very different from the typical leukemias and diseases that we would expect to see in children of those ages. And sadly, in a few cases, they ultimately were fatal. And so it did raise the question of, well, yes, the median age is 35 for onset of disease, but does that really mean that there isn't a risk, or is there a risk to the children in our cohort? Next slide. So we started actually by asking a more basic question: are the children in our cohort different than the rest of the cohort? And the answer is, for the most part, no. We looked at a variety of different parameters, hematologic parameters and clinical parameters. And the ones that jump out are the biggest graphs here. They are like the other patients in our cohort, have low platelet counts. The platelet counts are pretty comparable to the platelet counts that we see in adults, which are low, but not severely low. And so these are typically running in the upper tens to lower hundreds range. We also do see the hypereosinophilia in our children, especially including some who have absolute eosinophil counts over 500, over 1,000. Again, going along with the fact that we do see a lot of atopy and other autoimmune disorders in kids. And then when we look at a lot of the other parameters, which you can see on the right, and I won't go through them one by one for time, a lot of those didn't pan out, much like they don't really pan out for adults either. An interesting finding is, not unexpectedly, the children in our cohort do have elevated ISTH BAT scores, suggesting that they are having more bleeding and bruising than would be expected for age. But On the other hand, whereas almost half of the adults, both men and women in our cohort, have elevated ISTH BAT scores above the blue dotted line, which indicates normal, in children, it's only about a third, suggesting that if we are relying solely on phenotype for who we need to test, there is a possibility that we could be missing a very large, maybe even a sixth of our patients, just based upon this alone. And then it also shows that ISTH BAT, while useful and helpful, is not necessarily going to be definitively diagnostic of RUNX1 FPD in and of itself. Next slide. So to that end, we did actually look into the question of the heme malignancies. On the left side, we have all the individuals who we analyzed for this analysis. And for this analysis, we only looked at pathogenic or likely pathogenic. We excluded the VUSs just because we wanted a very clean picture of the patients who are at highest risk to see what that tells us to begin with. We have about 200 patients in that. They're evenly divided among male and female. Interestingly, about a third of the patients were in the pediatric 0 to 18 range, whereas the other 2/3 of the patients in this cohort are in the adult range. But yet, despite that, and despite the fact, about half of the patients that we identified having a leukemia or other heme malignancy were in that it happened in the pediatric range. So we also looked, and sadly, you can see there are several deceased children, but also deceased adults as well. When we look at the malignancies that we are seeing and break it down again between pediatric and adults, we see that the adults have the adult type leukemias, the MDSs, the AMLs, but the children, about half of our pediatric cohorts, about half of our cohort in the pediatric cases also have what we would consider more adult diseases like MDS and AML, and they also have some unusual ones like monocytic leukemias. When we look at the age of onset, though, and this is possibly the bigger point, is that what we do see is that there is a bimodal distribution towards the age of diagnosis. There is a peak at about 1 to 10 years of age, and then there's another peak at about the 40 to 60 years of age. And even the 40 to 60 years of age is probably a little bit lower than we would expect for the general population. And this is true both when we look at the patients who are enrolled in the study, which is the darker bars, as well as the patients who are just identified through the extended cohorts and looking through the family trees of all the patients that we have enrolled. And then in panel C, we can actually see the characteristics of the diseases that we have. I can't really go through this panel entirely just for time, but we do find as we see these malignancies scattered across a whole variety of different types of RUNX1 variants. And we see, as noted above, a variety of different malignancies. But what we also see is that the cytogenetics and somatic mutations, while present in pediatric leukemia, are considerably lower than what we would expect to see in adults. The adults, the vast majority of adults who developed an AML or other leukemia had 3 or more type somatic mutations, whereas in children, none of the children had that many mutations, and most were in the 1 to 2 range, suggesting that there may very well be a slightly different disease mechanism going on. Instead of an antecedent clonal hematopoiesis or something of that nature, it might be something of the more epigenetic variety. Next slide. Oh, sorry, and that is, and there is the evidence right there. So the next slide, we actually did then an analysis, a formal analysis of the risks of the disease across the lifetime, and what we see is pretty much exactly what we were seeing through our anecdotal data too, is that there is, as in panel A, you can see a decrease in the disease-free survival through about the first 2 decades of life. It tends to level out a little bit, and then it starts to pick up speed again at around 35 to 40 years of age. When we compare these results to the Fanconi anemia cohort that the NCI has, we see some interesting trends. When we look at the cumulative incidence, what we see is that Fanconi anemia, unlike RUNX1, has sort of just a generally straight monotonic increase in the incidence throughout life, whereas RUNX1 has that little burst at childhood, and then it takes off again later in life. And you can compare both of these are pretty bad compared to the general population, which is the black line along the bottom, and that's from the SEER databases. We can also look, instead of just the incidence, we can actually look at the overall risks. And we see that, again, RUNX1 being the red line and Fanconi being the blue, RUNX1 actually has a very high annualized risk of disease, about 500 per 100,000 patient years, as early as basically birth. Whereas Fanconi anemia, it starts off at a relatively low level and then slowly begins to catch up. Now, Fanconi catches up by about the age of 7 or 8, but RUNX1 remains relatively elevated compared to the general population well into the 20s and 30s. And even during that dip, the risk does not ever go back down to what we would expect to see in the general population. And so this results, as you can see in panel D, in about a 40-fold higher cumulative incidence by the age of 5 and a 50 to 60-fold higher incidence by the end of childhood for leukemias and the other heme malignancies in RUNX1 compared to the general population. And even into adulthood, despite the general population beginning to have more disease and then things leveling off a little bit in RUNX1, there's still about a 25-fold higher incidence of disease compared to the general population. Although by that point, Fanconi has now well outstripped it. And then final slide, please. And so really from this, what we are identifying is that, yes, indeed, pediatric patients really are at a much higher risk than compared to the general population. And it is a risk, at least early in life, that is comparable to other cancer predisposition syndromes, such as Fanconi anemia. And that really based upon this, as well as some very informative cases, early diagnosis and routine monitoring of pediatric patients with RUNX1 FPD-MM really is essential. And that hopefully, though, it will allow for early detection of heme malignancies and then more timely treatments. Okay. And we'll hand it over to BC from there.
Thanks, David. And so we've been getting quite a few questions about telomere length in the RUNX1 cohort. And thanks to the many patients that come out to the NIH yearly, we have enough data to answer some of these questions really quickly by just sorting through the data that we have. And even some of the other locations that study RUNX1 have also helped by sending some data to us. So the question here is whether or not RUNX1 FPD patients have short telomere, and the short answer, pun intended, is yes. Next slide. So why do we even care about telomeres? As you all know, it's important for genome integrity. And like telomere biology disorders, our patients sometimes can present with hypocellular marrow. We know about the predisposition to cancer. So I was able to look through about 40 patients, and I'll show you the data. These patients do not have malignancy. They do not have lung fibrosis, liver fibrosis, or some of these other phenotypes that we see in other telomere biology disorders. And I don't have the data to show you today, but we also know that these patients that I'm showing you do not have defects in the telomerase or sheltering complex, as you would see in other telomere biology disorders. So going on to the next slide. If you click one more time, I want to see if it'll build out. Okay, you can go back. So here I'm showing you a nomogram, and this is where about 830 patients, healthy individuals, had their blood donated, ages ranging anywhere from birth to about 100 years. And the telomere length was measured to create this nomogram that you see in the background. And I know you can't see my arrow, but these lines that you see, the blue at the 99th percentile, then the green 90th, 50th, 10th, and then the red at the 1st percentile, that's what I'm referring to when I say nomogram. So that's the background, and that's what it looks like in healthy individuals, 830 something to be precise. And the black dots are the RUNX1 FPD patients, and you can see just by glancing that our RUNX1 FPD patients are falling below the 50th percentile. And so they have shortened telomere lengths. And here you're looking at two different cell types. The top one are the lymphocytes and below are the granulocytes. And those were the two cell types that we focused on. The methodology to determine the telomere length here is FlowFish, where you would use flow cytometry to identify the cell types, so your lymphocytes and your granulocytes, and then FISH to actually tag the telomeres and enumerate the length. So on the next slide, here I'm showing you just the proportion within the cohort that have the shortened telomere length. And you can skip right to the red summary at the bottom. You can see that it's a high enough number. So for the lymphocyte telomere length alone, we have about 87.5% that have shortened telomeres. So that is referring specifically to patients that fall a combination of 1st to 10th percentile or less than the 1st percentile, which less than first would be very short. And that's something that you see very commonly in disorders like dyskeratosis congenita. When you look at the granulocytes, the number goes even higher, and now it's at 95% of the RUNX1 FPD patients, again, are falling within that 1st to 10th or below 1st for telomere lengths. Next slide. And so right away from the last slide, you can see that the proportion in the granulocytes is higher, meaning we had more patients with shortened telomere lengths when we're looking at the granulocytes. And so the next question I had was, is that shortened telomere length significantly shorter in the granulocytes compared to the lymphocytes? Because this has been seen in other disorders like aplastic anemia. And we can talk about why these differences arise later. But yes, if you look at the table above where I say all, meaning all of the patients, you see that the loss of kilobases for the lymphocyte telomere is minus 1.7, and for the granulocytes, it's minus 2.4. And this is significant, suggesting again that the telomere lengths in the granulocytes are significantly shorter than the lymphocytes. And then I break it down based on a 2-decade interval to see if there's a specific age group where it's more significant than the other, and that doesn't really pan out. And below, you can see the graph where 0 would be the expected median for those different age groups. And then you can see how much kilobases is lost. And you can see that for our RUNX1 FPD patients, they're all falling below the 0. And you can see the numbers that are falling below the 10th and the 1st percentile when you look at the lymphocyte or the granulocyte population. And you can see a lot more are falling below that 10th percentile for the granulocytes than the lymphocytes. Next slide. So then we thought, could we try to figure out, and this comes from a lot of the RRP meetings as well, as we hear you guys ask these questions, we asked, could we try to figure out when these telomere lengths actually start to shorten? And we were lucky enough to work with in genetic council out of California, who recommended patients to us. And upon the patients coming over, I was able to dig, and I found that they actually had over 9 years worth of data of telomere length. So it had been previously measured at that institution, and I could put it together. So patient P1 and P2 are related. They have the same mutation, and we'll go over that on the next slide. Patient P1, I have data starting at age of 2 years. The next dot is 6, and the last is 8. And then patient P2, I have data starting at the age of 12 and then 18 and 21. And the biggest takeaway for A and B is we're just showing you how the telomere length is crossing the percentile. So those of you that are used to pediatric checkups where a patient goes in for a pediatric patient goes in for head circumference or weight, you know, typically if a patient falls in the 90th percentile, they sort of track along that line. What we're showing you here is our patients are crossing the lines, again, suggesting that there's an accelerated telomere attrition. And C and D, we go to actually calculate how much of the kilobases they're losing. And the green line for both lymphocyte and granulocyte shows you that as you age, the loss should actually decrease. But for our 2 patients here, it's actually increasing, and their lines are going up as opposed to down. Next slide. So then again, questions that we're hearing is, for the individuals that I just showed you, if you look at both the lymphocyte and the granulocyte, the last mutation, so the arginine 346 proline a frame shift, though that's the mutation for the 2 patients that I showed you. And we were
1
lucky
minute
enough
till Q&A.
And we were lucky enough to have data on the parent. And so we wanted to see if the telomere length was significantly shorter in the offspring compared to the parent. And for the lymphocyte, you can, you know, maybe see that the parent is right between the 10th and 1st, and the kids fall below the 1st. But when we look at the granulocytes, they all sort of clump together. And then more importantly, when we look at other families, do we see a big difference? And no, we don't. Next slide. So then more importantly, we wanted to see how this one, first of all, in the table, the frequency of shortened telomeres compared to other inherited bone marrow failure syndrome. Right off the bat, for dyskeratosis congenita, you can see that those patients have a high percentage that are very short. So that less than first percentile that I showed you, where it's about 90% and above. But when we look at other things like Fanconi anemia or Diamond-Blackfan anemia or Schwann diamond, you can see that our numbers are comparable. And then below that, I have the graph to actually show you where the RUNX1 FPD patients fall. And you can see it's very similar to the telomere biology disorders and even fallen lower than some of the severe plastic anemia patients. Next slide. So then we just wanted to look at clonal hematopoiesis and see if it made any significant difference between the telomere lengths. The breakdown is what we are used to seeing, the B core, the TET2s, and no, we don't see a significant difference in telomere lengths in the presence of these mutations. Next slide. And then lastly, so our takeaway here is this appears to be a universal feature. So previously published work within the group characterizing the RUNX1 patients have shown you that, you know, a significant proportion, I believe greater than 90 have thrombocytopenia. We know that, we all agree. When we look at other things like hypocellular marrow, that becomes only what, like 17%? When we look at CHIP, maybe 30%. We have all these other factors that sort of fluctuate. Here we're showing you a feature that's about 90%, especially when you look at the granulocytes. So sort of a way to sort of identify our patients. But we have so much more to do. This is preliminary finding, but we brought it to you because we know it answers some of the questions that you've asked. But we're really excited and we have so much more to do, as you can see shown here. I won't read it in the interest of time and to also allow for questions. So thank you. Always a pleasure talking with you all.
Thank you very much, BC and David and Paul. Really appreciate you coming and sharing all this very interesting data. I'm opening it up to the group for any questions at the moment. I knew there would be some. Go ahead, Lucio. I saw your hand up first.
Thank you. And first, I want to thank the whole NIH team. I mean, from the community, seeing how this data is now emerging, looking at patient samples is amazing in the telomeres, in the platelet function, in every single aspect. I think this is a unique effort, and it's a very big effort. We appreciate that a lot. The question is specific to David is, you mentioned that it looks like the progression in pediatric cases versus adults is different. And do you see a difference in the first somatic mutation that may accelerate more aggressive or not the progression of disease being different?
That is actually an analysis I'm trying to do. I'm not sure we have enough data, despite the size of our cohort, to really give a good answer. It doesn't seem like at the current point, and one of the things I kind of had glossed over, there wasn't, or at least in the children, but even the adults, we haven't been really seeing a lot of recurrent lesions that seem to be predictive. One of the things I think the benefit of the cohort study that we have, and as long as things continue on their path, we'll continue doing it, is that we are now able to watch these patients longitudinally. We don't have as many pre-malignant samples, I think, as we would like to have. Sadly, as this cohort is matured, as as we accumulate more, we are starting to see more and more leukemias like occurring on study, and so that is allowing us to start to do a little bit more real-time analysis, as well as to have a much more robust pre and post malignancy set of samples. So we're looking at that, but right now we don't really see it. I think there are a couple mutations that maybe are a little more concerning than others, but it's really hard to say right now, other than there does seem to be a difference from that standpoint.
Thank you.
Thank you. Len, please go ahead. You were next.
Thanks. Yeah, I was just interested in the mechanism, and maybe I don't know this literature, but are telomerase type genes lower in expression in RUNX mutants, and does RUNX1 bind to telomere production genes?
So we know that telomerase is expressed in the hematopoietic stem cells, and then it goes away as the cells differentiate, and it could actually explain the differences we see between the lymphocyte and the granulocyte because they can reactivate telomerase. In terms of whether RUNX1 binds to telomerase or has any direct interaction, that's definitely high on our to-do list to see if there's a direct interaction or effect. But great question, something we're also looking to try and answer.
Right, but also we do have data on telomerase gene expression level, right, BC?
Yes, from the RNA-seq, and there was no significant decrease in expression.
Right, not only telomerase gene itself, but any genes related to the sheltering complex and telomerase complex. There is no change, and also no mutations in those genes.
It might be worth, we published some ChIP-seq data with RUNX1 and human CD34 cells. It might be worth just cataloging those genes anyway and just seeing if RUNX is bound.
Sure,
great. Thanks, Len. Vijay, you're up next.
Yeah, thanks. Just related to Len's question, BC, do you think that this could be not attributable to a direct effect on telomerase per se, but actually just because the stem cells are undergoing more self-renewal divisions? Because that's telomere length provides a great measure of self-renewal divisions. A number of people have used it to model clonality and things of that sort. So I was just wondering if you could maybe comment on that.
Yeah, thoughts. Paul and I argue about this all the time because that was my first answer was, they're exhausted and they're renewing a lot. The papers come out on inflammation and there are other disease models. But I don't know if CAT is on. I think we've actually done some of that with the iPSCs and we don't see that, right, Paul? So I think that sort of got shot down when I initially said it, but we're hoping to move on to models where we can answer those questions more technically.
Right, no, I mean, it's a very good question. I don't discount that as a contributing factor. I think it certainly is, especially considering the difference between granulocytes and lymphocytes. However, the degree and the persistent or even accelerating loss of telomere length is suggesting is more than that, more than the stem cell exhaustion to account for the severity and this widespread universal feature.
Yeah, just one comment though. It might not be exhaustion per se. It could just be the number of divisions that a clone has to go through to maintain a certain pool.
One way to get at some of that is also looking at telomere length and other tissue types in a RUNX1 patient, just to see if you see the same attrition or not.
That too is on our list. We're setting up the models to work on that.
Yeah. Okay, I think we can take one more. If you're okay, Vijay, on time, we can take one more question. Go ahead, Anupriya.
Yeah, thank you for the great talk. My question is related to Lan and Vijay.
You're breaking off, Anupriya.
Yeah, we can't understand your...
Can you repeat?
I was intrigued by the differences in granulocytes versus lesion, and like the telomere length is different in granulocyte versus lymphocytes. I was wondering, is it due to differentiation bias because RUNX1 FPD has more myeloid bias versus lymphocytic bias? And then I was curious if something like that has been observed in other disease model.
We didn't quite get what you were mentioning. You were talking about the myeloid bias.
I think she's asking about the difference between the lymphoid and granulocytes. I couldn't hear you clearly.
Yeah,
I think your question.
I'll type maybe. Yeah, that's what I was wondering, Roland.
Yeah, so actually I don't know if the figure is up in the summary. Well, yeah. So we think there are a number of different ways we can explain the differences between the lymphocytes and granulocytes. So I'll answer your second question first. Yes, this has been seen in other disease phenotype. There's literature on this for aplastic anemia. They see that same discrepancy between the granulocytes and the lymphocytes, where the granulocyte telomere length is significantly shorter than the lymphocyte. One thing that we allude to is that for the granulocytes, you differentiate to a granular site, but you have constant replenishment coming from the HSC. For the lymphocyte, you have division happening within the cell, and those cells can reactivate telomerase. So we think that could explain some of the differences, but we also have a number of other explanations that we're looking into that could explain the discrepancy. But yes, it's been seen in other disease types.
Great.
Thank you.
Thank you. Thank you again, NIH team. Really appreciate you sharing all this impressive data. Very insightful. All right, so we will move on to Dr. Vijay Sankaran from Boston Children's Hospital, who will talk to us about clonal dynamics in RUNX1 FPD. Take it away.
Great. Well, thank you so much for having me. And I'm sorry I haven't been able to join some of this meeting. I've been trying to, I'm at another meeting. I'm joining you from my hotel room. So apologies for not being able to participate more. But really terrific to be here. And my goal in the next 15 minutes is really to try to give you a very brief overview of work that's in progress. And really what I would love to try to make sure we facilitate and have time for questions afterwards, as well as discussion to discuss some of this ongoing work. So I think really the central question that's been driving the work that we've been doing is, we know from the elegant work of Nancy Speck, Kai Tan, as well as from Len, that there, across these different systems, both in mice and zebrafish, there's certainly defects with either haploinsufficiency of RUNX1 or knockout of RUNX1 in terms of the production of hematopoietic stem cells. And yet, in individuals impacted by RUNX1 familial platelet disorder, we have no sense of what the clonality is in maintaining hematopoiesis. And I think that's been limiting us in asking fundamental questions. And specifically, to what extent can that altered clonality potentially contribute to the progression that we do know occurs and is increased in likelihood in individuals with RUNX1 familial platelet disorder? And so we've been trying to address that through some approaches that we've been developing. And specifically, if one really wanted to understand the clonal behavior, there's really two fundamental aspects that we would like to tie together. First, if we're looking at any kind of evolutionary or genealogic history, ideally we can get some inference about the genealogic relationships between different individuals, different species, as suggested here from this original sketch of Darwin, or so forth. But at the same time, really to get a full picture, we really want to understand the behavior, whether of organisms or so forth. And so we believe that this is important in terms of understanding clonal dynamics and clonal behaviors, because ideally what we could do in terms of cellular behaviors is start to perform lineage tracing or clonal tracking to be able to deduce genealogic relationships between different clones, between different cells, to try to understand something fundamental about what are the relationships that are occurring. And moreover, at the same time, we would really like to understand aspects that we can at least measure within a cell, such as the transcriptome or the epigenome. And so that's really been the goal that we've taken on. And to be able to do this, this has been a longstanding goal that we've had broadly in terms of understanding human hematopoiesis. Several years ago, in collaboration with the Vijay's group, we actually found that after a lot of failure and a lot of attempts to identify good ways to track different clonal populations, that mitochondrial DNA mutations actually served as a valuable and potentially useful marker of different clonal dynamics to be able to track more closely related cells and separate those out from more distantly related cells. And we were able to perform a number of proof of principle experiments where we could take different clonal populations, perform barcoding, and at least at a gross level, we could reconstruct some of these clonal relationships through these kinds of approaches. But some of these early approaches that we had were limited by the number of markers we could uncover and our ability to really reconstruct faithfully many of the phylogenetic relationships that were present. And so this really motivated us over the last few years to think about approaches that we could use to improve upon this current paradigm. And this really led Chen Wang in our group, a joint postdoc with Jonathan Weissman at the Whitehead Institute, to develop this approach that we've termed REDEEM, or single cell regulatory multiomics with deep mitochondrial mutation profiling. And the essential development with this approach was that we could conduct a typical multi-omic approach using 10x genomics chemistries, but combine this with a fixation and permeabilization in whole cells prior to processing of cells, and then couple this as well to a enrichment of the mitochondrial genome to be able to more deeply cover it from the attack sequencing libraries. And we could also combine this with an error correction approach that really allowed us to more sensitively call mutations at lower variant allele frequencies. And this together allowed us to get really 3 layers of information. We could get information about the phylogenetic relationship between different cells from the mitochondrial DNA mutations. We could get RNA sequencing information to be able to determine gene expression in that cell. And at the same time, we could determine what are the regions of accessible chromatin within the same exact cell to be able to get insights into what's happening in the epigenomic state. And this approach allowed us to, in the context of healthy human hematopoiesis, deduce lineage relationships, be able to deduce aspects of clonality within these individuals. But importantly, we could also couple this with information that we could get on cell state. So we could get coupled information on gene expression and as well on accessible chromatin. And that could allow us to deduce what cell types were present, what was their behavior, what were changes in gene expression, and so forth. So those original approaches were obviously very exciting. And around the time that we were reporting some of these original approaches, we were very fortunate to team up with Paul and Gigi and Len to get this RISE award that really facilitated and catalyzed some of the early work that we began to apply these kinds of approaches to better understand RUNX1 familial platelet disorder. And so my goal today is really to try to give you an update on what we've done. And this has been tremendous work. Really, much of the credit goes to Jonas Gudera in our group, who along with Chen Wang, Michael Peschla, and Tang Gao have been both developing and spearheading many of the newer approaches that we're developing, which I'll briefly mention in the next few slides, but also who have been helping us gain critical insights into some of the underlying biology. So just to give you a sense, today I'm going to describe to you in-depth analysis that we've done from 4 individuals with RUNX1 familial platelet disorder from the marrow samples that we have of these individuals who do not have overt malignancies. And typically, we're assessing about 10,000 cells per individual with an attempt to deeply cover both the hematopoietic stem and progenitor cell compartment, as well as the broader bone and marrow mononuclear cell compartment, just to make sure that we can maximize coverage. So I want to give you a few views, and I'll couple this with information and data that we have from young, healthy individuals, as well as representative older aged individuals, typically in their 70s or older, to give you a sense of what the variability is in terms of clonal architecture. I would just note that these have all been jointly processed together, so all of the calling that we have of variants and so forth has occurred in a similar manner, and so this is really comparable. So in young individuals, as we've seen in some of our prior studies, we see this nice polyclonal architecture of healthy hematopoiesis with a variety of clones that can be called. We can simply reconstruct these phylogenetic trees as I'm showing you here, and I'll discuss some of our approaches to actually improve upon these our current calling approaches. In aged individuals, what we've seen almost universally is that even in the absence of detectable driver mutations, there's a breakdown in clonal architecture with typically an oligoclonal pattern that's seen. But in RUNX1 familial platelet disorder, really remarkably, there's a much more dramatic oligoclonal pattern that's seen, even if we account for the number of cells sampled and so forth. Now, this is certainly variable, and you can see, for example, that this individual RUNX number 4 certainly has a more polyclonal pattern as compared to some of the other individuals. But this is universally seen across at least the individuals that we've profiled to date with RUNX1 familial platelet disorder, really suggesting that there is a unique architecture clonally in RUNX1 familial platelet disorder. Importantly, we can couple this with information that we're getting on cell states. So I'm just sort of showing you UMAP projections where we've called different cell types that we've been able to call from these different clonal populations, including some of the expanded populations that you can see, as well as some of the other clonal populations. So we're getting gene expression information, as well as accessible chromatin information from all of the cells that we profile in these contexts. And I'm showing you some of these other individuals as well, where you can see that here there are a number of expanded populations, as well as some other populations that persist. And this really covers the full spectrum of hematopoiesis there. Now, one can look at this in different ways, and we could take this type of data, and I think I showed some of this at the last meeting, but really, if we use approaches like this Cassiopeia approach to really look at what are potential branch lengths and where might there be clonal expansions, in the majority of RUNX1 patients that we've examined, we see these pretty distinctive clonal expansions, as well as an oligoclonal pattern of hematopoiesis as compared to both young, healthy individuals, and even more pronounced than what is seen in aged individuals. So there does seem to be a propensity to see and see often multiple clonal expansions. But, and certainly we can also look at this in other manners. So we can look at this using nearest neighbor approaches. And I believe I've shown some of this as well at the last meeting, where you can see that there are often many more clusters of cells that are more closely related to one another, suggestive of the fact that there are these clonal expansions that are often seen in the context of individuals with RUNX1 familial platelet disorder. But many of the approaches I've shown you so far really rely upon earlier approaches that we had for calling mitochondrial mutations. And I would just stress that there have been limitations to this, particularly about the reliability of these approaches and the ability to rely upon markers that could drift in particular. And just citing the HHS secretary for the sake of it, so, while he was trying to describe children overburdened by mitochondrial challenges, as a pediatrician, I don't necessarily think that's the case, but certainly we in our lab have been overburdened by the mitochondrial challenges in terms of better calling many of the variants that are present. And so this is something that we've been working on. And in particular, we've improved upon our ability to really account for drift in mitochondrial mutations that might be present, since we can, of course, lose mitochondrial genomes, we can gain new mutations, we can see random drift of these. And by accounting for this using Wright-Fisher population genetic models, we can actually start to call more confidently those phylogenetic trees or branches that we can call with greater confidence as compared to those with lower confidence. And I illustrate that where we, for example, here have taken healthy donor human hematopoietic stem and progenitor cells, label these with lentiviral barcodes, which serve as our gold standard, which are colored across this row here at different colors. And then we can use mitochondrial mutations, as shown below in this heat map, to reconstruct the phylogenies of these. And for each of these branches, we can call a confidence with different cutoffs that we've colored here with this red, white color scheme. And this allows us to start to tease apart branches where we have greater confidence in calling the clonal relationships from those where we don't necessarily. And by using a trimming approach then, based upon this branch confidence, we can start to reconstruct phylogenetic trees, as I'm showing you here on the right side, really illustrating how, by improving our calling approaches, accounting for some of the drift of mitochondrial mutations and really accounting for the quantitative degree of a variant being present or not through its variant allele frequency, we can gain a lot more information and really be able to better call clonal dynamics. And we've certainly benchmarked this, but I won't go into this for the sake of time. So if we start to do this across young, healthy donors, we once again see the same pattern where we can see a polyclonal architecture. We can start to take phylogenetic trees or branches of the phylogenetic tree that we're more confident in and separate those out from those that we're less confident in. And we can see in older donors, of course, there's this oligoclonal pattern with clonal expansions, as we've seen and numerous others have seen. What happens in RUNX1 familial platelet disorder? Well, once again, we can start for the confident branch to start to see this oligoclonal pattern with pretty significant clonal expansions, particularly those that we're most confident in. So I think we're quite confident in the fact that there's both this oligoclonal architecture that's really maintaining hematopoiesis, but also critically these pretty distinct clonal expansions. And I would just mention, with what BC had mentioned in the last presentation, certainly there is the suggestion that if you start with fewer stem cells, many of these stem cells might need to actually undergo more self-renewal divisions in order to compensate and make up for the lack of stem cell clones. Now, as we've started to put this data together, of course, you might be wondering, well, we have all this other information on cell state or on gene expression or accessible chromatin. So can we start to integrate this? And so we started to integrate all of the data we have from both young, older individuals, as well as individuals with RUNX1 familial platelet disorder. We call a variety of cell types. I've just labeled them here for simplicity with their overall kind of cell states. And as you can see, we have nice coverage both in individuals with RUNX1 familial platelet disorder as well as individuals unaffected with this condition to be able to compare all of these different cell states. And we've started to conduct some preliminary analyses on these, and I'll just report on this really briefly. So if one just simply asks, what are the differentially accessible chromatin regions in hematopoietic stem cells or molecularly defined hematopoietic stem cells? Interestingly, and consistent with a lot of the discussion that's been happening here, we start to see a pretty extensive increase in interferon alpha and gamma responses in the accessible chromatin of these regions. And this is just really looking at global gene sets, but certainly I think that we need to do much more analysis of these. But these are really in these specifically molecularly defined hematopoietic stem cells. We know from the elegant work of Nancy Speck that really in the context of deletion of RUNX1 within the GMP compartment, there's an increase in inflammatory signaling that seems to be mediated by ERF and STAT transcription factors, and that this then results in a cascade of interferon signaling. And we also know from Anupriya's work that in RNA sequencing data from bone marrow of individuals with RUNX1 familial platelet disorder, similar pathways are also upregulated. And we've been having wonderful collaboration with Nancy to try to look at this more in the GMP compartments. And indeed, in the accessible chromatin that we're looking at in the GMP compartments, we see upregulation of TNF alpha signaling, reactive oxygen species signaling, interferon gamma, and so forth. And so certainly, I think we're starting to get some interesting insights on molecular changes as compared to the healthy donors having the same exact cells or those unaffected with RUNX1 familial platelet disorder. We can see interesting changes in MEG progenitors, and I just mentioned this in passing. For instance, we see increased DNA damage repair and some other pathways there. And similarly, even in monocytes, we can see that there's upregulation of a variety of inflammatory pathways, interferon alpha and gamma and IL-6 signaling that are all present here and upregulated. And I would just note that even if we compare this to aged individuals, the degree of inflammation that's seen in RUNX1 familial platelet disorder seems to be much greater in many of these populations, really suggesting a unique inflammatory signature that's seen there. So I think we have a lot of ongoing work that we're still working to do. I think our hope is to continue to profile additional patient samples, and in particular to try to look longitudinally, because I think one of the advantages we have is by marking many of these clones with these mitochondrial markers, we can follow them. Ideally, we would love to examine what happens in the context of progression to leukemia in these individuals, and how this occurs, what clones give rise to this to better understand some of the progression. And I think that as I've suggested to you by kind of the gross global analysis I've shown you here, that by starting to examine in different subpopulations and even different clonal populations, differentially accessible chromatin regions or differences in gene expression, I think we can start to get some insights into what's happening within a cell that might be either extrinsically mediated or intrinsically mediated, and try to get insights into some of those pathways. And ultimately, our goal really is to try to use this and other approaches that we're trying to develop to ultimately track what happens in the context of RUNX1 familial platelet disorder that both sets the roots of acquiring malignancies in these individuals and increases the likelihood, but also how this progression might occur and how we can better understand it and potentially intercept with the process. And so with that, I just wanted to finish up by acknowledging the people who were involved in this, most notably Jonas, who's really led this work with Chen, Michael, and Teng, as well as a number of other people in the lab. Particularly, I just wanted to acknowledge our close collaborators, Paul, Gigi, Len, Jonathan Weissman, with whom we've been developing many of these approaches, Nancy Speck and Daniel as well. And I really wanted to acknowledge our funding sources, but most importantly, all the individuals and families impacted by RUNX1 familial platelet disorder who've really made this kind of meeting important and impactful and possible. So with that, I wanted to thank you all very much, and hopefully we can have a few minutes for questions. Thank you.
Great. Thank you so much, Vijay. Beautiful work as always. Please go ahead and ask your questions if you have a question. I have a question. With the 4 patients that you have analyzed so far, did any of them have CHIP, or do you have that information at this point?
I believe we do, and maybe Paul can speak better to it. I mean, we did try to assess patients who typically were on the younger end. We know our age range in the patients we've studied is 25 to 55, and but I, you know, in terms of the detectability of CHIP, I'm not sure we have we've fully analyze all that information, but I think that there's certainly, you know, both cases of detectable clones and not. I would just note from other work that we've done, for example, in aged individuals, and we've looked at a large number of aged individuals, we've detected clonal expansions even when there aren't detectable drivers, and we know that there are many drivers that we haven't necessarily been able to detect. So I wouldn't necessarily totally feel, you know, I guess I'm always hesitant to say we do know or don't know a driver at the moment, but certainly that's something I think we need to look at more carefully.
Great. Any other questions? Wenbin looks like has a question. How is FPD patient 4 different from the other 3?
It is the youngest patient we've examined. So of all the patients, this is a patient that's in their 20s. So perhaps this is age related. I don't think we know, and I think that this really motivates us to try to better study what's happening. I would say that there's certainly, though, even in comparison to age-matched individuals who are unaffected by RUNX1 familial platelet disorder, there does appear to be more oligoclonality, although not necessarily the same kind of clonal expansions. So I think that there's much more to understand there, but this is the youngest individual, at least, that we've profiled.
Got it. Thank you. And it looks like Yash is also curious about whether the inflammatory signature that you found in the HSCs, and I think it's getting back to this question about driver mutations and whether if you were able to separate out whether some of those inflammatory signatures are just innate because of the germline RUNX1 mutation or could be exacerbated by additional mutations.
Yeah, I think we have more work to do. You know, we've really looked at this at a very gross level. So everything I've shown has really been, if you look at molecularly defined HSCs using, you know, our integrated measurements of cell state, how do those compare to young, you know, to healthy, unaffected individuals with RUNX1 familial platelet disorder? So I don't think we can comment much on are there particular clones that behave one way versus another and so forth. Having said that, I do think what we're seeing is suggestive of the fact that globally, the hematopoietic stem cells are showing this inflammatory signature, and to a much greater extent than, you know, and we sort of intentionally compared even those monocytic populations where you have a large number of cells. This is seen, you know, even to a greater extent than what we think about, you know, with inflammatory changes with aging, you know, which people often bring up the term inflammaging. And, you know, even comparing that, you know, this is, you know, with a log 2 comparison in the scale I showed you, there's quite a dramatic shift. So I think we probably have to dig into it more. We have to look at more of what's happening at the clonal level. Yash, to answer your question, but I think that globally even, I think we're just seeing a global shift across all the cells.
How does this affect your thinking around therapeutic interventions? I mean, because we're talking about so much change in sort of a variety of inflammatory processes.
Yeah, I mean, I hesitate because I don't think we've done the right kind of studies, but I don't think we know what's causing this, right? These are just observational studies and we have data. You know, I would say, you know, and this partially reflects maybe my obsession, but I think that to the extent we can, whether looking at telomere length in bulk, there was just a nice paper published earlier today from Trevor Graham's group about looking at bulk DNA methylation as a measure of clonal dynamics, at least in cancers, and certainly I think it could be applied in the blood system, and they've done some work on this. Using such markers, I think, will be critical, because I do think that in our field, we probably need to think beyond just blood counts, beyond just the standard measures that we have, and just think about clonality. And I think that's where, we're not going to be able to do redeem on that many individuals in the clinic, and it's not going to. But I think as we start to gather this data, our hope is that we can correlate that with some measure that's more easily readily done. And I actually think that'll be an important surrogate to try to look at, because I don't know, and I think we have to gather the data. I think that's why we were really motivated to try to understand what happens longitudinally in individuals, how these clones progress, how this evolves, and so forth. But I do think we need to understand this, because I think unless we understand that, we're going to have trouble intervening in the way we might like,
Mm-hmm. Yeah, I appreciate your thoughts on that. Any other questions? And we can certainly have a bigger discussion while folks digest the talks we've had so far this morning and afternoon. So we now head into a break. Thank you, Vijay, and again, NIH team, for great presentations in this last session. And we'll have a break until 12:45 Pacific, 3:45 Eastern. And then please come and join us at that time for a patient story. Virginia Monk is sharing her life story and the impacts of RUNX1 FPD on her and her family. So we have a nice video there, and then we'll end with our last two speakers, and of course our group discussion at the very end of the day. So please join us again at 12:45 Pacific, 3:45 Eastern. See you soon. I'm delighted to now transition to a very special part of our program today, which is to hear a RUNX1 FPD patient story. And this story is about a woman named Virginia Monk, who is very near and dear to our community. She's been with us for years, and we're grateful that she took the time to tell her story. And I think you all will find it very interesting, but also inspirational, and hopefully make you feel really good about the work that you're doing, and that you have the potential for impacting people like Virginia and her family. So with that, I will have Catherine go ahead and play the video.
I grew up in Sydney, Australia, on the harbour. Lots of beaches, lots of sailing and surfing. And right now I live on the west coast of British Columbia on a little island called Galiano Island, just off Vancouver, near the San Juan Islands. My grandfather, his name is Virginius Henry Randolph Murray. He would have possibly inherited the title of the Earl of Dunmore. The men in my ancestral family inherited this Scottish title, but then they'd be the Earl of Dunmore, and then they'd die prematurely, and it would have to be transferred to either an older son, if there was one, or to go over to another part of the family, a cousin, a nephew, as long as their last name was Murray. So there was quite a record of these men dying of some kind of blood disorder. And in fact, an interesting thing is I go back to the famous or infamous last British governor of Virginia, who was John Murray, Earl of Dunmore, who was kicked out. But my family seemed to like that name, and I'm Virginia. My grandfather's Virginius, my aunt was Virginia. But because of that, we know that there was blood disease in the family. I developed psoriasis when I was 8, and my mother also had that, and she had arthritis, and she sort of knew there was some connection to inheriting illness. And I think as a protective thing to us children, she said, oh, people who in the family who have psoriasis, they don't get blood cancer. So she wanted to kind of believe that and have hope, as we all do, that her children would not experience the cancer. And she didn't really. She had anemia and blood transfusions sometimes, but she didn't, as far as we know, have blood cancer. And then the family stories that were pretty big were that she lost her brother. He was 17, and her father at 63. My first cousin died of ALL, and he was 32, and his daughter is a survivor from ALL as a toddler. She's still going strong today. So we were aware of this thing in my family, and it was not until there was the familial cancer study in Australia that I joined that they were looking for the gene. Is there a gene for this? And then once it was discovered, then, oh, this is what's going on. And then once the gene was found, by then my younger son, Gabriel, was probably around 2005 or 6, maybe. He and I decided to be tested, and he wanted to be, he was now an adult, young adult, and he decided he also wanted to be tested for the gene, and we both turned out to have the gene. Living with RUNX1 it's interesting because I think in my case, learning about it after I've lived a lot of my life and done a lot of sort of adventurous things that I didn't care, I didn't give a thought to, that what if I got sick suddenly and I'm in the Andes in South America, where I did do a lot of traveling, or I'm in India, so that's one thing. And I think because my mother didn't succumb to leukemia, I just put it out of my mind. Once I had the RUNX1 diagnosis, I was eligible within our medical system to get blood tests every 6 months. And so I just did that, didn't pay too much attention. My platelets are always a bit low, but that's normal for me, and life just went on. My son, Gabriel, who at the younger age learned about this, he bruised really easily, and that became obvious that this was caused by having RUNX1. When he left high school, he became a carpenter. He did the full training, and he also did a lot of travel. He packed in a lot of really interesting adventures in his young life. He also took up working with stained glass. He did beautiful designs. I was away visiting family in Toronto when he had to go into hospital and was diagnosed with leukemia. He sent me a text to let me know, and he did really short texts, and the text said, "So I've got leukemia." And I have to say that one word, "so," kind of says it all to me. Like, yeah, he was thinking this might happen. He was really clear on wanting to be tested, and even when he then got the diagnosis of leukemia, there was a huge amount of hope and belief in the medical system, and he, in October 2019, he went to hospital. He had a whole lot of chemo, and went into remission, which was needed for him to be able to have a stem cell transplant. And so in January of 2020, I remember when early part of that time was the discussion about having the transplant. His brother offered to be a donor if the markers were suitable, but Zach's blood wasn't suitable enough. So we were assured there'd be a donor registry person, which indeed there was. And when Gabe was interviewed by his doctor, he said something like, well, I'm really looking forward to seeing what happens with this. And he was part of a big experiment in his life that he wanted to see how it would go. The stem cell treatment is incredibly brutal. And he was a strong young man, and he came through that, and he persevered with everything. He was willing to, do what it took to get well and to live. Once the leukemia came back at about the 20-month period after the stem cell transplant, or 22 months maybe, and it was T cell that came back, and I think he'd been mostly treated for B cell. That was a different time. There was hope that there'd be remission again and he would go home, but that couldn't happen. He died of leukemia, and I think the death doesn't mean he thought it would happen and therefore it happened, but I do think he packed in a lot of good stuff in his life, knowing he needed to live his life really to the full. A child getting sick and a child dying is the most difficult thing, which I live with, right? But then I didn't have a worry for my own RUNX1 situation until I finally was diagnosed with MDS. The concept of hope is so vital for keeping yourself going. And I would say that even though I've discovered that my particular MDS, perhaps because it's germline, I don't know, it does not respond to the traditional treatment. Now I've got the diagnosis of maybe only living for up to 2 years, which is really, can't get my head around that. But I still have some hope by participating in what I can do for research. That's one thing. And so, although I'm not going to travel because of my immune system to NIH, that is there as something I could do possibly. I was able to send blood samples, which was amazing. And sirolimus is the current one we're looking into. I haven't, not that I can be in the study at the moment because I have MDS. My husband is, it's difficult for him. This is a different situation to the two of us losing our son. It was his stepson, actually, and my son. Fortunate enough to have his other two sons are my two stepsons. So I have a lovely supportive family, which is great for both of us. And we like to see friends, which keeping community is also very good. And we both like that. So we meet people for dinners and gatherings, but small gatherings. So it's important. It's wonderful to have him in my life during this. I've found not getting too involved, but doing something proactive about understanding your condition. But be careful not to get go down rabbit holes on Google or spend too much time on it. And but do keep up with what's going on. And I guess if you live in the States, go to NIH if you have that opportunity, because it's another way to just keep tabs on yourself and take advantage of this amazing research that which needs to be done more and more on germline people. And find something that can replace azacitidine for people with MDS or whatever is needed for RUNX1 patients.
Well, I hope that story was as moving to you as it was to me. Virginia Monk, is someone who I think has incredible strength, and I love that she's choosing hope and choosing to be proactive, and I think surrounding yourself with community is great words of advice. Oh, so thank you. Thank you very much. Oh,
She's
you're
here,
with
actually.
me.
Hi, Virginia.
You're here. Oh my gosh, welcome. Thank you for being here, and thank you so much for sharing your story.
Yes, I'll just say hello, and I really appreciate doing this video. It's wonderful. Thank you all.
Well,
Thank you, Virginia.
Yes.
Thank you for all you're
Thank you,
trying to do.
Virginia.
Yeah. Mm.
Good luck. All the best.
Thank you.
Virginia, I just want to say thank you also. You've come across as someone with so much wisdom and strength. You obviously are, don't just come across. And I appreciate you sharing your story. Thank you.
I appreciate what everyone is doing for this. Yeah, it's amazing.
Thank you. Well, we will get there together. All right. With that, I'm going to transition to our next session. It will be led off by Dr. Bobby Bauman from the University of Pennsylvania. And this is a session on what I called, it sort of tried to encapsulate the two next talks, which is uncovering leukemia transforming factors. So please go ahead, Bobby, take it away.
Thank you. Does that share okay? Can you all see that okay? Yeah,
We
great.
can see it.
And yeah, my appreciation for that moving video and this kind of research community as a whole. It's as being as a bench scientist, this is definitely one of the most inspiring research groups I've ever been a part of, as far as reminding us why we do what we do and who we do it for. I'm going to jump into some of the ways we think about modeling this syndrome and this disease that people live with. And forgive my kind of slow transition into that after our kind of moving video. I want to introduce the way we think about kind of leukemia as a whole, and particularly how we've begin to think about RUNX1 mutant disease. So for us, leukemia is a clonal evolution process. This means it's something that changes over time, and I think that's something we can all certainly appreciate at this conference. This is a population of cells that can have some heterogeneity early on, right? And throughout the course of life, new mutations or selective pressures may lead to the expansion of different cells or populations. And we know that this is not always a linear process. Sometimes there are many different mutation acquisitions that can occur. There are inflammatory pressures that we've talked about repeatedly over these past 2 days that can drive the expansion of different cells. But underpinning all this is a long process that also occurs, and we need to figure out ways that we can study it quickly, so that we aren't continuing to tell people that it's going to take a long time. So when we thought about how we wanted to approach studying cancer evolution several years ago, we thought we needed methods by which that we could really validate specific mutations that were found in genomic profiling as therapeutic targets. We wanted to ask questions, mechanistic questions about some observations that maybe some mutations were found late versus early. We wanted to be able to actually develop models to kind of order those mutations. And then we wanted to understand what happened to cells after, maybe if you had a perfect drug and you could eliminate a mutation, what would happen to that cell? And just really understand how cancer cells survive therapy, and understanding that disease evolution continues beyond just a disease initiation, but evolution continues throughout the course of therapy as well. So those are some of the major questions we had. I kind of want to introduce the different methods my laboratory has used to study those, and then I will kind of give a deep dive on one particular branch of that. We've looked at this through single cell DNA multiomics. We saw some of this earlier. We think that's a really important way to understand the clonal dynamics that occur. One of our biggest focuses has been in adult AML studies, where we've constructed a series of inducible leukemic alleles, so we can actually model these mutations that occur late. And we've been able to use kind of different synthetic biology tricks to turn on mutations and then turn them back off. And one of the things I don't often get to talk about, but we're going to focus on a little bit here, is how we've tried to model things in cell culture to try and capture these really long phenotypes that occur in mice, and try to understand how we can evaluate methods that are, sorry, evaluate mechanisms that underlie clonal hematopoiesis or mechanisms that underlie clonal evolution in a cell culture format that's actually, adjustable to perturbation, right? This slide was already introduced, so we'll just briefly say, and this has been mentioned, that the one interesting facet of RUNX1 clonal hematopoiesis is the differential abundance of different mutations that are not normally as abundant in age-related clonal hematopoiesis. And primarily here, we're going to focus on the B core mutation. That's something that we've been interested in modeling in conjunction with Nancy Steck's lab. The other large frequent somatic event that we want to keep in mind is loss of the second allele of RUNX1, and this is a very common event in both sporadic and germline RUNX1 mutant AML. And I share these slides with full knowledge of the people who generated this data, both in the previous slide and this one, are members of this community. So we're are in a good spot as far as understanding some of the genomic features of this disease evolution. And I think we're in the right position to continue efforts to model it accurately for both the early clonal evolution events and the late ones in leukemia. So with this knowledge in mind, we need to figure out ways that we can model clonal hematopoiesis and transition to leukemia in a time-efficient and scalable manner. And I say that because some of these phenotypes can take a long time, and in many instances that isn't a luxury we have. We need to be able to move faster in this field and how we're doing it. So I'd like to introduce the one method that we've gone through, and I think quite a few labs now have actually started to use this method or used it previously. So for some people, you are very familiar with these cultures, and for others, this might be the first time you've seen it. So maybe we can kind of show you how we've been modeling this both in age-related clonal hematopoiesis and our efforts to do it for RUNX1 mutant disease. So for some context on this, keeping hematopoietic stem cells and evaluating hematopoietic stem cell function in tissue culture has been a very challenging concept and a challenging method to employ. I'd say we've seen major advances over the last 6 to 8 years on being able to keep hematopoietic stem cells in culture and have them still look like stem cells a week later. And that time period is important because it gives us enough time to genetically manipulate the cells to test hypotheses or to deliver drugs so we can test their efficacy on killing or causing differentiation for these cells. We've tried a bunch of methods, and one that I think we've seen a lot of productivity out of are these bone marrow endothelial cell co-cultures. These co-cultures use a layer of immortalized endothelial cells that represent one of the endogenous niches of hematopoietic stem cells, and it lets us perform long-term cultures in this setting of mouse hematopoietic stem cells for pretty scalable culture, lets us do genetic and chemical screens. So we've done this, and this is some published data about just on alleles that we've looked at in adult clonal hematopoiesis. So some of the same alleles that we would be interested here in RUNX FPD, but just in a RUNX wild type background in this setting. And I think it's interesting because you see that, we looked at 4 different mutations here, whether it's in DNMT3A, TET2, ASXL1, and if we look at their competitive growth compared to wild type cells in these cultures, they all kind of win, right? These mutations cause cells to grow better or more abundant. But if you start to drill down into specific hematopoietic stem cell compartments, you can see the alleles start to have different effects there. So a DNMT3A knockout, for instance, will or loss of function will lead to expansion of hematopoietic stem cells, but the ASXL1 phenotype does not. I think we've learned these lessons in mouse modeling over years, and it's given us ideas of how these mutations may affect stem cell fitness. What I'd like to stress here is this is only a week of culture that we're able to recapitulate something that takes 4 to 5 months in vivo to identify. So, I don't think this is necessarily the same, certainly not the same kinetics that we see in vivo, but it does make these assays fast and amenable to perturbation. So, just because they're fast doesn't mean that's a good thing. We need to make sure that they actually are functional. So one of the most important parts about these cultures is we can take the cells after culture, transplant them back into mice, and see that these cells are able to transplant and carry along these phenotypes. So we're able to take these cells, culture them, perturb them, and then get in vivo readouts that are high fidelity to a stemness phenotype. We know that these cultures also have fidelity to genotypes that matter. Or drug treatments that matter. So one comparison of interest with TET2 clonal hematopoiesis. We can look at the ratio of TET2 versus wild type cells in these cultures and see that as the cells become more differentiated, this is where that fitness advantage starts to play out. So the LSK stem cell population, TET2s may have a slight advantage, but it's really starting to manifest more strongly in the more differentiated cells. And that gives us an idea of at which cellular compartment these cells are actually gaining their fitness. And critically, what we can do is take drugs like decitabine or an azacitidine analog and show that in an expected manner, the TET2 cells are more responsive to decitabine than the wild type cells. And this is something that in vivo or in patients can take many cycles to see an effect. And we're able to see it in just a week. So the scale of these cultures let us look at more than just one drug. We've done some work where we've looked at a series of different drugs at different dosages and and then looked at their ability to either expand or suppress a TET2 mutant clone. Just the scale of it is not quite a high throughput drug screen, but we could certainly do on the order of 60 to 80 to 100 different compounds to look at fitness of different molecules. In this prior publication, we then extended that to doing kind of domain-focused CRISPR screens. And in this setting, we have done about 300 different epigenetic factors to understand what the genetic dependencies were, for these different genotypes. And by and large, you can see that different genotypes have different dependencies. So we think this is a strong discovery platform to understanding different genetic dependencies. And there's a number of labs that have, employed these screens and really kind of drilled down into the RUNX setting. So I won't spoil too much of what some of those results are because Ben might cover them, or we will all continue to talk about them in slides or weeks to come. We began to do some of these cultures with RUNX1 mutant cells. We first started by looking at responsiveness to different cytokines. So here we have either wild type cells or the R188Q point mutation. And we can see that, there are more LSK stem cells with SCF treatment. If the cells seem more responsive to GCSF and FLT3 ligand as far as upregulation of the LSK compartment. And then what we've done, and this is data actually from Nancy Speck and Suan Fontenard, is we can use these culture systems to do competitive cultures between either, in this setting, a RUNX1 mutation with B-core or without that B-core mutation. And over the course of the culture, you can start to see this double mutant clone start to grow out and dominate the culture. So we think that these cultures are fast and have an ability to elucidate phenotypes that take a long time to see in vivo, and they're amenable to perturbation. One of the perturbations we were interested in is different cytokine treatments here as well. So this is a setting where we broke apart either wild type, and this saying RUNX R188Q, B-core loss alone, or the RQ B-core combo. And we see a stem cell like expansion in the RQ B-core combo. This is not a super robust expansion, and that's something we're trying to kind of work on and keep in mind how we can really model the kind of clonal synergy between these two mutation pairs. We are kind of moving this colony towards an SCL-CRE-ER, which we saw earlier today with the RUNX TET2 model, and we're going to be looking to see if we can kind of look at clonal fitness in the setting of an inducible B-core loss as well, in addition to loss of the second RUNX1 allele. We have had some previous preliminary data in collaboration with Nancy and Suan that had nominated menin inhibitors and perhaps a Hox gene signature. And unfortunately, this is negative data, but I thought it was important to share, at least in our hands. When we looked at the RUNX B-core mice, or here in this column, or the RUNX mice alone, we were hoping to see that treatment with Syndex 5613, a menin inhibitor, would suppress the RQ B-core mutant clone. And unfortunately, we just didn't really see much of a signal here. So we need, some new kind of discovery-driven methods to understand new targets for this common co-mutation, and two, see if we can really amplify the fitness of this RUNX B-core clone, because I do think we have a bit of a kind of dynamic range issue. We know it's a very common combination of mutations, as Paul and his group showed earlier, but the models we have so far, these clones seem to be growing relatively slowly, which might reflect the biology seen both in experimental models and individuals, but from identifying therapies that can actually suppress the clone, we need it to grow and expand fast enough to be able to actually knock it back down. So I think I have a couple minutes left. I want to take that time to highlight how we're moving forward to do that and what methods we're employing as a lab to try and kind of accelerate this disease phenotype towards a leukemia so that we can identify therapies that might be relevant even in the interception phase before that leukemia forms. We are repurposing our CRISPR screen approach here. So instead of looking for genes that are dropped out, we're looking to see what can synergize with RUNX or BCOR to actually cause larger clonal expansion. So these cells are in mice. We're pretty early in that, and we should get some results soon. This is kind of giving you a small sense of the mutation space that we're looking at. So things that we think do co-occur frequently. We have other guides for NF1, for instance, which I think would be both an interesting representation of MAP kinase pathway activation, but also speaks to that interesting case report we saw earlier today. So we'll be using this as a kind of clonal outgrowth screen to see which mutations can synergize and might help expedite some of the very laborious mouse modeling that is, I think very important, but maybe this can help give us our kind of refined signal on which alleles we can think about crossing. So right now, this is in a single guide setting. We'll have a RUNX R182 mutant mouse. They've received a single guide. I think we are going to be heading towards combinations most commonly. Our most leukemias, either in the adult AML space, or we think even coming in the RUNX FPD space, are going to have more than one mutation, and we can use this combinatorial guide RNA setting as a way to credential different combinations very quickly. So in this setting, and I think this is a useful and important method we've been using, you can express 2 guide RNAs in pairs, and then read out which guide RNA is there, which genes are lost by reading a barcode out. And this is done in an approach called a crop-seq vector. And there's a small technical detail here that, in the bigger picture may not be super important for people considering these studies might save you a very large headache down the line. So we'll kind of share how we're approaching it. Putting these guide RNAs in a crop-seq vector places them in the long terminal repeat of the lentivirus. And this lets you actually just pull out the guide RNAs with any poly A selection for single-cell RNA-seq or for RNA generation. So just a simple repositioning of where the guide RNA is makes the access to these guide RNAs and their barcodes significantly easier to extract. So this is something we're approaching now to try and clone these different combinations, specifically focused with a focus on BCOR and all these other cooperating genes. We'll read them out by bulk sequencing as well as single-cell RNA-seq. We've deployed this in the lab in the context of adult AML, so I'll just share some of our results there. We've used it in this setting for kind of clonal barcoding, so we can see which clones are expanding, and then get a sense of what their transcriptional profiles are to understand why we get expansion. So in that setting, we're taking our leukemic cells, we're infecting with this lentiviral barcoding library or this guide RNA library, and we're getting very good clonal representation. So we think we're going to be able to pretty readily look at 50 to 100 different clones in our culture system. And that should represent a decent fraction of this combination space. And I don't have much concern in our ability to scale this to cover thousands of clones. It's just going to matter how many we can afford to sequence. So we've looked at this and see great reproducibility between the replicates, which is always reassuring. And this is also to highlight that these cell cultures, they contain a lot of different cell states, right? So we're going to be able to not just say this is a lineage negative HSPC, we're going to be able to say that these cells are either maybe more monocytic or more dendritic or a progenitor state, and then understand how these mutations influence those cell states. All right, so I kind of leave that here, that these are our approaches by which we're trying to kind of rapidly identify which combinations of mutations are going to give a leukemia that's going to represent what's seen in the RUNX BCOR mutant patient population, and try to get us to kind of high fidelity model. Our kind of gold standard approach would be then to engineer those mutations back into either a mouse, so we can not lose context that this is a germline mutation and the RUNX1 mutations in the stroma and non-hematopoietic compartment are completely critical, perhaps, to the disease progression. So this is a kind of a representation of the library of alleles that we've generated over the years or received from other groups. We're looking to kind of build into it. So if you have a gene of interest, or maybe we should all kind of collectively think of, if we were to build a new TET2 allele, and wanted to combine with loss of B core or loss of RUNX1, is there kind of an optimal recombinase to place it under the control of? For instance, if we wanted to separate a TET2 mutation from loss of the second allele, it might be better if we had a flip-inducible TET2 loss or a GRI-inducible TET2 loss. And my lab certainly has the expertise to characterize and generate these, but I'd much rather do it with people who are actively studying TET2. We can build these things together and make sure that they're useful for everyone's experiments. When we have systems like this where we can control the different mutations at different times, we're able to do really interesting things biologically. So here's an example of something we've done where we're able to, in an adult AML setting, model acquisition of either NPM1 or FLT3 and able to switch the order of these genes and ask, did it matter to disease development? So these methods for being able to induce these different recombinases are ready to share. They're useful. And if anyone's interested in kind of taking genes they're working on and figuring out a way that we can kind of expand this ecosystem towards alleles that are useful to the research community, we would love to do that. So please let me know.
1 minute Q&A.
Perfect, thank you. So in summary, we think this culture system is a really robust way for us to be able to look at clonal outgrowth in a short period of time. We've used it successfully to kind of look at dependencies in IDH2 TET2 mutant cells, and we do see some synergy with RUNX1 B core, even though that synergy is pretty mild in vivo. So I think our biggest challenge right now is figuring out ways to accelerate that synergy. And I think by learning how we can make this, pair of mutations progress towards leukemia, that will unveil ways that we can stop it in the end. So I just want to thank the people who've done the work. You guys heard from the undergraduate in our lab, Aditya, who worked hard on some of the transcriptional profiling this summer. Eileen Klein's new clinical fellow lab, who will be taking up this work, and then Angela Yoon, who's off to med school now, has previously worked on this. We've had very generous funding both from Alex Lemonade Stands and via RUNX RRP and as well through the ACS. And just grateful for the opportunity to present and share our work with you, and would be happy to discuss further. Thank you.
Great. Thank you so much, Bobby. Beautiful. And I'm really excited about the system that you've generated. Looks like we have another question for Anipriya, from Anipriya.
Great work, Bobby. Somehow I can't turn on my video. It's saying disabled, but I'm here. So I was very curious about your in vitro culture method. That's the way to go if we want to do things faster and get the initial read on the phenotype of those secondary mutations. As you see from our work and Esther work, the immune cells seems to be also playing a role.
Yeah.
Is there a strategy to have those cells in these culture method to have like more cell-cell interaction impact?
I think we can definitely make the cultures more complex. That's not a problem. It kind of comes down to source of cells, but I think those are also, isolatable. We have not made any efforts towards evaluating like antigen presentation or T cell function or anything like that. And that's just really not, I need some expertise and a collaboration for that. But we've also looked at mesenchymal stromal cell co-culture. I feel pretty confident we can spike in a lot of different cell types and see how they affect the culture. Len's brought up macrophage spike-ins. We can produce macrophages from the cells we lay down, but I don't think there's any concerns in the ability to spike more in as well. So I would feel pretty comfortable with that.
Yeah, cool. And just clarification, these are het models or
Yeah, so we've done the TET2 hets and saw a similar but less robust phenotype. The DNMT3A, this is the knockout. We've done the R882H and seen interesting phenotypes. And the ASXL1 here is probably the one I feel probably least comfortable with, because I'm not sure I totally understand how ASXL1 works, but this might in the end be not the best ASXL1 model because it's a complete knockout and not the frame shift that we'd probably really want to actually be modeling.
Thank you.
Thank you. I had a quick question, and then Paul, will go to you. When you generate the bone marrow endothelial cells for the ex vivo culture system, would you anticipate that there could be a difference in the way the cells behave if you used a germline RUNX1 bone marrow endothelial cell?
It's a great question. I think this came up last year, and we should really do that. We haven't yet generated. So these endothelial cells are an immortalized endothelial cell line, right? So we should certainly try and immortalize an R188Q. I think that's something we can definitely look into. And I think this sets you up to have niche interactions to evaluate. It hasn't been our primary interest, but understanding how they talk to each other, I think this would be an interesting platform for it.
Yeah, absolutely. Okay, thank you. Paul.
Yeah, essentially similar question. So just wondering if these are all using mouse cells, right? Have you tried? You think these could be used for human?
Can I punt to Wenbin or ask if he can answer? Because Wenbin and I have talked about this a lot, and he has better data, experience, and knowledge of the human than I do. So sorry to put you on the spot, Wenbin, but you know better than I do.
Maybe we'll wait for after Wenbin's talk.
Yeah, well, I mean, I can briefly discuss this. We have been using a humanized system to expand the leukemic cells and also human CD34, from cord blood, peripheral blood. It's working, but it's not as robust as the murine system.
Okay. Thank you.
Okay, we can take one more question. Go ahead, Lucio.
Yeah, and I was surprised about you in picking the SCL-CRI system to do this, and I understand that that system is pretty ineffective, and it varies depending on, as any CRI system depends on between locus and locus. So how are you going to do screenings and assess for whether you're dealing with a heterozygous or homozygous setting in the gene?
Yeah, it's interesting because I've heard people say that it's heterogeneous, and maybe we should share. I know we've talked about dosing. We've seen this to be one of the most robust CRIs I've ever worked with. And maybe we are overloading the mice with tamoxifen, but we don't see a lot of heterogeneity with it. And we dose over the course of a week 8 milligrams of tamoxifen by oral gavage, and we're getting really excellent recombination. So at both loci and at reporters, right? So from a screening standpoint, I don't think actually we need the SCL. We're going to rely pretty heavily on lentiviral guide RNA. So sorry, from a guide RNA screening standpoint. From the CRISPR screens we've done, we've sorted reporter positive cells and confirmed by excision PCR that this is greater than 90% effective. We just don't see as much header. I mean, it's not the FGD5 CRI, and Nancy can speak to that and Suan more than I can, but I think that one's very ineffective. But in our hands, SCL CRI has been an excellent CRI to drive high recombination rates.
With tamoxifen, you have to balance between toxicity and efficacy.
Certainly. I mean, tamoxifen is going to be myelosuppressive. I fully understand that. We've really, for the entire screens you see and everything that was in this prior publication, we dosed mice with TAM at 8 milligrams. 2 weeks later, sorted reporter positive cells. And, I understand there's potential caveats, but all the phenotypes kind of recapitulate what you'd see. So I feel pretty comfortable with the system.
Great. Thank you. Thank you, Bobby. All right. We're going to thank you for a great presentation. We are going to transition to Dr. Wenbin Zhao from Memorial Sloan Kettering Cancer Center. Please take it away. Sort of a late breaking abstract. We found out last minute that you could share your data. So welcome. Happy to have you here. You're on mute, just in case you didn't realize.
Okay. Can you hear me?
We can't.
Yes, but you're in presenter view.
Oh, presenter view. Okay.
Yeah.
How can I switch?
You just switch at the top.
Top. Yeah, swap screen.
You're good.
Good. Okay. Thank you, Katrin, for inviting me to give this talk. First of all, I'd just like to say, I'm very grateful to the foundation for supporting me as an early career investigator a few years back. I mean, during the COVID time, but it seems I survived. My research focus is pretty much on RUNX1 and germinal RUNX1 condition. And let me see. Okay, so I don't need to say too much about FPD. Just saying that this patient has a very high risk for transformation to MDS and AML and rarely AIL2. It's roughly 40 to 50% of patients eventually progress to minor neoplasm. I know the NIH cohort, as Paul just today said that they start to pick up somatic RUNX1 mutation in patient progress to MDS and AML. There are a few cohort published from Anna Brown and also recently from a European Netherlands group showing that when patient progress to minor neoplasm, the most frequent somatic mutation is RUNX1 itself. It up to almost 50% of those patients will have second RUNX1 mutation. If you count AML, it's even higher, suggesting that the two hits on RUNX1 is essential for the leukemic transformation. And then you have other frequent mutations seen in AML too. For example, NRAS, KRAS, FLT3. In a recent Netherlands group, they said about 24% of those IPD AML harbor FLT3 ITD mutation. So this leads us to basically set up a model for leukemic transformation in FPD. Basically, we propose that if we cross the RUNX1 genome model, which provided by Lucia here, and then with the flux allele, and then on top of that, we can lay over the FLT3 ITD allele created by Bobby. So that's the leukemia model, mouse model I will focus on. And then Bobby and Nancy's lab are working on a alternative FPD AML model as shown here. Basically, heterozygous RUNX1 mutation with B core, and then later on at a PHF6 to address that. The other probably a subset of patients did not show the second RUNX1 mutation. And I know Lucia's lab working on RUNX1 and NRAS, though those models also show the AML progression with a latency about like 6 months. So first of all, we cross the RUNX1 genome RQ allele with flux allele, and then evaluate the phenotype of those mouse model. Basically, the RQ allele function as a loss of function allele. As you can see, RQ knockout mimicking homozygous RUNX1 knockout in terms of thrombocytopenia and lymphopenia. And then by using those allele crossing with FLT3 ITD, and then on top of that, we have flipper ERT2 to induce the flipper recombinance, and then we also cross with NHGRI to basically knock out the second RUNX1 allele. So after this complex crossing, we obtain the bone marrow cell from these mice, and then transplant into CD45.1 congenic recipient. And in the first few weeks, mimicking a germline RUNX1 mutation FPD condition. And then, you confirm the engraftment, you start to give PIPC to delete the second RUNX1 allele, then you have a homozygous knockout condition. And then you can confirm the complete deletion of the second RUNX1 allele just by check CBC, because once you have a second RUNX1 allele deletion, you have severe thrombocytopenia, and then you can give tamoxifen to induce the FLT3 ITD allele. So once you give tamoxifen, those mice start to develop leukemia, and all the mice died within 3 months after ITD activation. So it's a very permanent AML model. And as you can see here, the c-KIT positive cell in the peripheral blood, and also the blast in the peripheral blood and the blast in the marrow. So we ask the question, how can we systemically identify dependencies in FPD or FPD AML? We know that, from FPD to FPD AML, there are some somatic mutation occurring, but we don't know how it works. So we designed this system that we are using the CRISPR screening, using different library, and to do the screen at a different stage. We have epigenetic library, which, Bobby's initial paper used that. And then we created inflammatory laboratory. We also created kinase laboratory. So we start to perform CRISPR screening at a different stage. As you can see here, the first epigenetic screening at the FPD stage has been completed, and the other stage is still ongoing or partially completed. So today I'm going to show you the epigenetic screening data. So here's basically that's the system Bobby just mentioned. It's a BMAC culture system, and this has been very successful in the lab. The beauty of this system is that when you use this system to do CRISPR screening, you not only identify just one gene, you identify the pathway. So that happened to Bobby's initial paper, and then that also happened to other people using different allele. And those pathways are different, depending on what allele you are using. For this study, focusing on RUNX1, as you can see here, many candidates are H3K4 methyltransferase. As you can see here, ML4, ML2, ML5, SADD2, those are all H3K4 methyltransferase. And because ML5 is a shared dependency among all the genotypes, so we decided to go for ML5 first. And we use an independent guide and validate, independently. So we ask, is this dependency also, works in vivo? Luckily, we were able to get a conditioned ML5 allele from a collaborator in Germany. 3 months before his lab closed, he retired. Anyway, so we obtained this allele and we crossed with RUNX1 RQ flux allele. And then, as you can see here, it's clearly that this dependency also works in vivo. If you knock out ML5 homozygous, you basically reverse the thrombocytopenia, and the leukopenia pretty much is a B-cell lymphopenia here. And when you look at the megakaryocyte morphology in the bone marrow, as you can see that in RUNX1 knockout or RQ knockout, the megakaryocyte, normal megakaryocyte is absent. But if you knock out ML5, you basically reverse back to normal. So we also asked whether this dependency function in the FPD AML model too. And we crossed this FPD model with ML5 homozygous. And we basically did the same induction. As you can see here, after tamoxifen administration, the leukemic arm all progressed to leukemia, but ML5 knockout arm, none of them die. Actually, some of them still develop leukocytosis, but they don't die. They do not have leukemia. Those mice showed leukocytosis is pretty much a FreeZ3 ITD phenotype. So here showing you the comparison between the leukemic infiltrate in the left, and then on the right, without ML5, the leukemia infiltrate in the liver is nearly all gone. So we start to look at the transcriptome, and here, as you can see, if you do k-means, you get two clusters. One is the genes are downregulated in IQ knockout and reversed in the absence of ML5 knockout. Those are enriched for B cell program. So we know that in mice, RUNX1 is required for B cell development. And then we are more interested in the second cluster because those genes are upregulated in RUNX1 and then reverse back to wild type in the absence of ML5 knockout. Those genes are enriched for megakaryopoiesis and staminase. And again, as Bobby pointed out, there's a Hox gene upregulation here. And there are lots of inflammatory gene upregulation, including many RF family. We start to investigate how RUNX1 interact with ML5. We did a ChIP-seq experiment, and here you can see this is the ML5, also called KMT2E. So in wild type, you can see a clear RUNX1 binding peak, which is absent in IQ knockout. And this is at the promoter region of KMT2E. Interesting thing is that, in the absence of RUNX1 knockout, H3K4 transmethyl, which is an active mark for promoter, and H3K27 acetylation, also active mark for the promoter. So both are upregulated in the absence of RUNX1, suggesting that RUNX1 probably directly suppress ML5 expression. And at the RNA level, indeed, when we compare wild type and the RUNX1 IQ knockout, ML5 is upregulated in IQ knockout, not other ML molecule. 1, 2, 3, 4, they have no change. So what is the ML5? Initially thought one of the ML family, H3K4 methylation transferase, as shown here, ML1, ML2, 3, 4, and 5. But later, people found that in the PhD domain and the SET domain, those are different from other ML family. And the second, if you look at this gene, it's much shorter. Other ML is more complex. Third, the SET domain has no intrinsic enzymatic activity. So it's now people basically lump this with some other H3K4 transmethyl method transferase, different from other ML family. Very little is known on ML5 function. Although it does not have set, the set domain does not have intrinsic enzymatic activity. But if you knock out ML5 in the cell, the H3K4 transmethyl level is dramatically down. That has been shown. So ML5 probably regulate H3K4 transmethyl through some other mechanism. And we look at the H3K4 transmethyl in the wild type IQ knockout and IQ knockout, ML5 knockout. As you can see here, so H3K4 transmethyl, again, there are two clusters basically. The first cluster is H3K4 transmethyl is down in IQ knockout, but the other is up. Most of genes are up. And then those are, again, basically return back to a wild type level in the absence of ML5, suggesting those H3K4 transmethyl in those genes probably are dependent on ML5. So we are working on to figure out the downstream mechanism. And here's the working hypothesis. In wild type condition, RUNX1 pretty much function as a suppressor on ML5 and many other genes. But in the absence of RUNX1, the active transcription factor or some other complex come in to activate the ML5 expression, and the ML5 via some mechanism contribute to H3K4 transmethyl and then lead to downstream gene expression, which will lead to I did not mention here. So the HSPC differentiation is pretty much biased to megakaryocyte. And then in the absence of both RUNX1 and ML5, you basically, you don't have much H3K4 transmester. So those genes activation is still downregulated. Okay, so in the last two slides, I just want to show you a very interesting technology we have been using in the lab. As Bobby pointed out, it's essential to deliver the recombinants into the cell for disease modeling, and he has been pioneering those orthogonal recombinants delivery, which if you make a mouse model, it takes a lot of time to do it. And when you start to cross with 3 different recombinants into one mouse model, it's very complex. So in the lab, when Bobby was still in the lab, we also used the other ways to deliver the recombinants. For example, electroporation. It's working, but it's highly variable. In a good day, you get a 30–40%. In a bad day, you get a 5% efficiency. So we try to tackle this problem by looking at other method of recombinants delivery, and we focused on lipid nanoparticle. And as UPenn group published a landmark paper, I think in Science in 2023, showing that yes, the LNP actually can deliver many things in vivo, but you need antibody conjugation. You need a conjugated with c-KIT antibody for the targeting. And without a conjugation, the efficiency is very low. They use a serum-free condition. So we tested that. Yes, if you don't have serum, the LNP cannot deliver things. Yeah, the basically we use a LNP encapsulate GFP mRNA. So without serum, the efficiency is almost 0. But with serum, it's very efficient, and the procedure is so easy. You just like add a cytokine, just add a drop of LNP to the culture, and then you just wait. And then because we know that in order, for stem cell expansion, you cannot have serum in the system. So we try to find a serum replacement, and we were told that human ApoE4 actually works as good as a serum, which we confirmed. Yes, with ApoE4, you don't need a serum. This will give you same efficiency. And the beauty is ApoE4 won't kill the stemness, with 1 or 2 week culture. So we also transplant those cells in vivo, and they have long-term engraftment. And then we start to test. We work with the company, made those customized CRI flipper recombinant LNP, and as you can see, it's so efficient. Yeah, where you only need 0.5 microgram LNP mRNA, and you can get 100% efficiency. And for flipper, I mean, for Korean G is very efficient. For flipper, the efficiency is not that high. We use a reporter called FLTG, which has a TD tomato and a GFP. So if you have a flipper, you will activate the TD tomato. And if you have later you add a CRI, you are going to activate the GFP. But since we use MS CRI, so it has, it's well known, the MS CRI has leakage. So that's why even you give flipper, we start to see some GFP here. But if you combine this, it's about 40% at the best for flipper. But for Korean juice is 100%. Since the flipper is less efficient, we want to test whether we can use this way to model AML. So we have the NPM1 3 both FRET earlier, which can be activated by flipper recombinants.
1 minute till Q&A.
Okay, and then we just add LNP flipper mRNA to activate this allele and transplant it into the mice. As you can see here, with 4-HT, that's the traditional way to do it. Those mice all progressed to leukemia and died within 10 weeks after transplant. And with flipper LNP, those mice all die, but it's slightly delayed. But you do get a very similar AML with similar phenotype and also pathology, and they almost the same degree of leukocytosis, although it's delayed. So that's all. And again, thank you for the support from RUNX1 research program, and we are recruiting a postdoc. If anyone has a candidate, let me know. And thank you for all the collaborators listed here.
Thank you so much, Wenbin. I know you've showed me the data before, but I'm still so impressed by how MLL5, loss of MLL5 can completely reverse in vivo, the platelet effects and even correct the megakaryopoiesis as well. It's a striking result. I'm curious, and maybe you showed this and I didn't catch it, but have you ever looked to see in individuals, whether it's somatic, sporadic AML or MDS that have RUNX1 mutations, do you see upregulation of MLL5 in those AMLs, MDSs? Like, do you see that this is happening in human tissues, in human disease?
Yeah, we haven't looked at that. I look at the mutation. There's no ML5 mutation. ML5 amplification occur in some cancer, but I don't know the RUNX1 mutation status. Like breast cancer, OB, it's called endometrial cancer.
Mm-hmm. Okay. Would be interesting to see if you see that.
Agree.
Lucio, please go ahead. Your hand was up first.
Well, being very nice data. And I imagine that you're thinking about this. Are you planning of trying to validate MLL5 results in human cells to see whether some of the markers of FPD are reverted?
Yeah. We are working with Paul to see whether we can use the iPSC system to basically knock out, CRISPR out ML5 and see whether, I don't know, I mean, I don't have much experience with iPSC, but the one thing I think we could do is to differentiate those cells into megakaryocyte to see whether, by knocking out ML5, you can restore the megakaryocyte maturation.
That would be a good way to do it.
I was just going to say that Morty Ponce has done a lot of that type of megakaryocyte differentiation from human-induced pluripotent stem cells. So, and he's got a RUNX1 line or two at least.
Wow.
So you might want to reach out to him. Yeah.
Okay.
Dirk, you had your hand up next. Please go ahead.
Yeah, very nice talk. Can you hear me?
Yeah.
Okay, great. So nice presentation. I have a technical question. So I'm really intrigued by your data with the human ApoE4 and the LNP delivery. I mean, this looks really amazing.
Yeah.
So as I was wondering, maybe you mentioned it and I didn't, I missed it. Did you try this in different, highly purified cell populations, or what was the cell type that you actually worked with for these experiments?
Yeah, so we use the lineage negative depletion. So it's lineage negative enriched cells. The concern is whether it has a like a preference on certain population or has toxicity on certain population. We look at the HSPC compartment after 1 week compared to non-treated versus treated. It did not change too much. No, I mean, as far as we can tell, there's no difference, and
So-
compared to 4-HT as well.
So that means you're seeing also the CRI induction or the GFP, depending on the experiment in more purified populations.
Yes, yes, it certainly can get into LTHSC.
Okay, yeah, very cool.
Yeah, thank you for sharing that. Any other questions for Wenbin?
Have you tried the Cas9 LNP?
That's next step, yes.
Okay. Just curious.
Yeah, I mean, ideally we can use this for CRISPR screening.
Yeah.
And which probably, if this works, will help the human CRISPR screening too.
Right.
Absolutely.
Exactly. That's great. All right. If there's no further questions, we can start our group discussion. We're running a little bit behind, so our discussion will be shortened a little bit, but I think that's okay because we've had lots of great Q&A discussions thus far. Nancy, do you want to kick us off with the first question, or I can? It's either way.
Oh, well, I like the question about where we see the most immediate opportunities for collaboration across labs. So is there anything people have that you would like to get your hands on, or people you would like to work with?
I love it.
I was expecting everybody to say they want Ben's
Yeah,
LNP.
I'll kick off. I like to test Bobby's system using primary patient samples. If you want to do it fast, why go to mouse cells? Let's do it, with human cells.
Yeah, absolutely.
If it's not available, let's develop it.
Yep, I agree. I mean, I think we have some work to do to make sure it works before we touch the precious samples, but we can do that work. And I think Ben, you've done some of this already, so I think it's also good to just try some of the new solvents that have been used too. And for those of you that are stabilizers in cell culture. So the other thing we didn't talk about was polyvinyl alcohol cultures, but Soluplus is another reagent, looks to be particularly good for human cells. And we know that we can combine polyvinyl alcohol with the BMEX to kind of get best of both worlds. So that's something we haven't tried yet on human, is combining some of the stabilizing agents with the BMEX for human. I don't know, Wenbin, if you guys have done any Soluplus or PVA for that, but there might be a nice venue for that, and doing it with real patient samples I think would be excellent.
Yeah, we played with PVA and Soluplus in the HUVAC system. Yeah, that's the human equivalent of BMAC. It's not as good as we expected. I mean, when we transition to human, it's always difficult. But we are working with the collaborator in Shaheen Rafi lab, because they made a new line which actually overexpress NKX transcription factor. It seems those new line actually is almost 10 times more potent than the old line. So we'll see.
By the way, for those of you who don't know, Bobby and Wenbin collaborated extensively when both were in Ross Levine's lab, and now they're both, along with me, actually on an ACS LEAP grant. So continuing to work together.
Yeah, I was going to add, I think Bobby system, even with mouse cells, is very powerful. If we can model other cell types, and if we can use like some of our single cell RNA-seq data, for example, to screen more inhibitors in co-culture model, I would love to take that an advantage. And if we can model this clonal competition, I think we still don't understand how these inhibitors impacting clonal competition. I think that would be really cool because mouse model takes forever. We can definitely recapitulate later in mouse model, but I think we can use for these new interventions, clonal competition, in vitro, long-term culture, even mouse model. I know human system has lots of challenges.
And on a I think that's one reason why, I know we're very early days on what Aditya showed earlier, but why we want to get some of that rapid low pass RNA sequencing done, so we can screen through many different inhibitors or combinations and then deep dive on maybe a single cell for the ones that have the best signal, right? So we've been brainstorming ways where we might be able to do these co-cultures instead with human cells, whether the HS5 stromal cells that will certainly support mouse, and in that way we might be able to actually disambiguate mouse and human transcripts and still don't have to purify cells in it. So getting through a high throughput rapid manner, I think, can get us to signal a little quicker.
That's
Yeah,
great.
so I also, I mean, I just made a note here. I mean, I would love to try Bobby's system and also Wenbin's LMP system with, you know, with our imaging modalities when we, you know, look at single cells, how they behave dynamically over time, measuring different ROS species to measure, you know, DNA damage in living cells. I don't know if people remember this, but that's what we are doing and interested in the context of RUNX mutations. So I think there's so many applications I can see. We should really talk about, you know, if you're willing to share these tools with others, that would be really great.
The
Absolutely.
LMP's platform technology is so easy. You can just send me an email and I will send you the protocol. And if you need an adequate of the reagent, we can share that too. Because when you order, you need to order a bulk. But we have those adequate to share.
All right, that sounds good. Yeah, I'll definitely do that. Thank you for yeah, offering that.
Yeah, thank you, Wenbin. Any other thoughts on systems, platforms that people want to get their hands on? Speak now.
I think one thing I've been struck by is there's a lot of great cytokine measurement data that's been presented. And, you know, on the pre, you've had some of these. We're always interested in this, but I could definitely use some advice and expertise in the best way to get these multiplex ELISAs done, whether people are doing them in-house or if you like a place you're actually shipping them to, or which assays. It seems like strong functional data that I think we could do a better job of characterizing our models on this cytokine production. So if anyone can raise your hand, let me know if I should email you, and I'd really appreciate it. But whoever feels like they have great cytokine data, I think that's something we could be analyzing better in my lab.
Great, thanks, Bobby. Any other thoughts or comments? Okay. Well, I always like this question, and I think I asked it yesterday too, but I think it's helpful to hear from all of you what you thought were perhaps more surprising results or results that will certainly impact the work that you do now after hearing what you heard today or yesterday too. You can include yesterday as well. What kind of shifted your thinking or maybe influenced how you might run your next experiments or think about your projects? I'm gonna wait until someone says something.
Well, I can say something.
Okay.
You know, Vijay and I have been talking. It's very interesting that he has this clonality data, and it's very similar to what we saw in our fish. I mean, pretty much the same thing, that you have fewer clones, and some dominate, but fewer clones. So, you know, one question I've thought about is whether there is a bottleneck in the patients. And that bottleneck, you know, leads to a premature clonal expansion, maybe as you're heading into teenage life. And it's during that bottleneck that you end up getting second mutations because your progenitors or stem cells are trying to catch up. And there might be a therapy which would increase the diversity of clones of stem cells in the patients, which might have to be used developmentally, like when you're a teenager or something like that. But you know, we have one compound that increases diversity of clones, this RO5 compound that works through RUNX. So I don't know if it would work in the patients or not, but compounds that would increase diversity might prevent the mutations from actually happening later.
Len, can you help me understand how you increase diversity after stem cells have emerged?
Yeah, so you basically can create a situation where the stem cells divide one or two more times after they're born. And when they divide, they're never exactly the same. So that population ends up essentially becoming the bone marrow clonality. So if you can, increase diversity, I think when you're a young child, over a period of time, it might make you more resistant to, stresses, chemotherapy, or radiation. And also, if you have a RUNX mutation, it might make you more resistant to getting a leukemia.
How do you think that fits with the data that David Young showed today, where we see this bimodal risk of hematologic malignancies? And do you think that clonal diversity could be impacting in the pediatric setting? Because
Yeah,
it's like,
yeah, that's the way I think about it, is maybe there's a deficit of stem cells in the germline RUNX mutation. And so they're trying to catch up and start, expanding. And so you end up getting pediatric, leukemia at some point. And then maybe the same process repeats itself much later, as we heard from the patient. And so, maybe this is the way it works, is you're monitoring your stem cell clones, and if you're not diverse enough, you have a problem. I don't know what other people think about that. It's a hypothesis more than reality.
Yeah, I think a decrease in clonal complexity is my favorite hypothesis for what clonal hematopoiesis is, and that RUNX is an extreme case of that, whether it be through an early first hit, but more likely through what you said, and what I think Nancy and others have shown, that there is a decrease in some production of LT-HSCs or HSCs from birth. So if you're already bottlenecking a small population to begin with, I guess the herring part of this, and there's good support for this, that means all those mutations are happening, really, really, really early, right? And all CHs is unmasking these clones through removing. It's a denominator issue, right? And that's kind of how I've come to think of it.
Yeah,
but then there's that expansion issue, right? So I wonder if, at least in Vijay's data, it looked like then all of a sudden there's these specific clones that are just expanding like crazy. So it's almost like they're not even using the diversity the limited diversity that exists is also not being.
Well, one way of thinking about that, Katrine, is there's pressure on the system, low diversity. So then some clones may acquire mutational things that are undetected, or epigenetic changes that make the clone dominant.
I think it would be really interesting to know if that decrease in clonal diversity happens really early, like in the, umbilical cord blood, I think would be a really interesting place to start looking to see if already at birth the diversity is low. And that seems like something the foundation maybe could help organize.
Yeah, I think we talked with since the last meeting, we talked with Paul and Vijay trying to figure out how can we get our hands on umbilical cord. And I think, Paul, where we left it was just you had to make sure that via the IRB at the NIH, you could actually access stored umbilical cord, because we do have individuals in our community that have banked umbilical cord for their children, and they're just sitting there and they want to be able to release it. But I think that was the next question, was whether you could actually take those in, right?
Yeah, but also I think our discussion initially was whether save those cells for potential therapeutic purpose. And we were saying, it's probably not the best idea to do that. But for research purposes, I don't think we have much issue with that. But Natalie has a recent paper on the pregnancy of patients, IPD patients. So she's probably have a better grasp or knows the possibility of the maybe which patient, which family may be expecting, and then we can, because this is really you need to arrange to really to get the samples for this kind of study. I think it's feasible.
Yeah.
Natalie, you want to comment?
I think from a regulatory standpoint, we're totally fine to bring in samples. I think our concern was just that families know that like once we take the samples, they can't use them in the future if, they would be helpful. So I think we've told a lot of families on the study, like, please don't destroy them. If you don't want to pay for storage anymore, like, please send them to us. But, I think we want to be very careful and making sure families are fully informed before they donate those types of samples.
Mm-hmm.
So, yeah.
So related to the clonality, one of the things that I thought when I heard the talk was to try to link whether the clonality is a consequence of the inflammation or not. So and that's testable, and it could be tested in cells or in some models to see whether if you reduce inflammation, if you induce more inflammation, whether you change the clonality.
Yeah, I mean, I'm favoring that macrophage model where the macrophages help the proliferation of the stem cells. So basically, it might be, things like IL-1 and TNF-alpha that Annapurna has told us about, or it could be something more about the intrinsic macrophage or monocyte behavior that's doing it.
Yeah, I've been thinking a little more about the shortened telomeres. It was great to see that data. And, with the conversation which we were just having, whether this really indicates that the stem cells may be undergoing more divisions, it'd be nice to try to measure that somehow, if we could.
Yeah, I agree with you. I think that to me was one of the eye-openers. Now thinking back, it seems to be obvious or expected, but I think it's one of the most novel findings that are coming out from the patients recently. And what is causing it, I think, is going to be critical to know. And as far as I know, using mouse models is a terrible approach because the way telomeres are maintained in mice is different than in humans. We know that from very old results that people have been trying to test that. And so I wonder, I don't know which one would be a good model to test, because in vitro systems also are stressors. So I don't know if that would be the best system to test for telomere.
Yeah. So along this line, Len, how about in zebrafish?
Yeah, I was going to say. So,
what's the telomere length in zebrafish?
Yeah, so telomere length is exactly like humans. And I have a graduate student who's been working on a TERT and a TURC zebrafish telomerase mutant, which you only have to wait one generation for to get the phenotype. Those fish develop cytopenias, and we've been looking at their clonality, and there's definitely clonality issues with the telomerase mutant that really are pretty similar to the RUNX mutants.
Right. I mean, can you measure telomere length in zebrafish?
We measure telomere length with
Great. Why don't you do that in our RUNX1 knockout? Because anecdotally, we didn't publish it. For those mice and those fish, they live beyond 1 or 2 years. All of their kidneys are bone marrow failure. They are very, very low cellularity. So we were just at that time we thought, well, they have bone marrow failure, but we didn't go analyze further. We thought, well, who knows what's exactly going on. Now retrospectively, that's probably related. So please go ahead and measure.
That's a very good idea, Paul. And I was thinking the same thing during the telomere talk. The defects that my graduate student is seeing is very similar to the RUNX mutant. And it wouldn't be wrong to think kind of what Nancy was saying, that there's a proliferation that's happening because you don't have clonal diversity. So and those telomeres come into effect at that moment, So I think it's very reasonable. So we'll do that.
I think so. Short telomeres also can stimulate some inflammation too, through DNA damage responses and all. Whether that could be related at all.
There literature on that, Alan?
What's that?
There are literature on that short telomere
I think,
relationship with
let me look.
inflammation.
I'll have to look. That was my impression, but
There's, believe I also saw literature. Is anyone familiar with the literature on transplant outcomes and short telomere length? I thought I remember maybe it was Coleman Lindsley who published something in MDS and short telomere lengths. The shorter the telomere length, in the recipient, the worse the outcome and the increased risk of poor outcomes. Does anyone remember that? I'll look it up if no one remembers it. But yeah, that I thought of that and I thought, oh gosh, that's not necessarily a good thing if that's true, if there's some kind of mechanism there.
Is Leah still here? Leah should know. Maybe Leah is an expert on transplantation.
Yeah. I'll do a quick search and see if I find it too.
Sorry, I couldn't unmute myself, but yes, you're exactly right. That data is out there.
So something for us to think about when we think about transplant again and the risks of transplant.
We have a question here from Laura Babbitt. Are there any thoughts on the new research on vitamin D supplements and telomere length?
Laura, I don't think this group has the expertise in telomere biology, but I know a group that is very well connected to the whole telomere research group, the Team Telomere. They fund research in telomere biology disorders, and we could find out what the latest is on vitamin D, unless someone on this group is aware of that data. I'll get back to you, Laura. Any other further thoughts on telomere length, or anyone want to contribute thoughts on other results that they felt were surprising or could influence some of their work back at the bench or in the clinic? Seems people are starting to get tired.
Well, how about we get to what gaps or challenges remain that we should address?
I like that.
And prioritize in funding. We're treating you all like an enormous SAB.
Yes. There's so much wisdom and knowledge here.
Yes, there is. Len just turned off his microphone.
Well, I'm going to ask a question to you, David, and I think I know I asked this to you before, but I think your data is really important for our patient community and for clinical care. And I'm curious about, now seeing these high rates of heme malignancies in young patients, what you believe should be the recommendation. I know, again, this is, one study, but that's what we have, and we have to work with what we have. What is your recommendation to patients and families about monitoring for hematologic malignancies?
Well, there's some that's in the data, and there's a little bit of anecdotal material that we've found, some of which is actually in the paper itself, just there wasn't time, honestly. But I think to a certain extent, it really points to the importance of a couple things. I think the most important part, I think it's a two-way tie here. I think one of the two most important parts is that you need to get diagnoses early, especially in patients or in family members. Once the family member is diagnosed, the cascade testing, I think, needs to be done. Not emergently, it's not like rush to your nearest doctor, but it needs to be done, and it needs to be done in a timely fashion to establish diagnoses for the rest of the family, especially children. I think one of the things that's come out of it is that while the phenotype is important, and this is actually a conversation that came up in a slightly different context, we would say that penetrance is pretty close to 100% in RUNX1 FPD, but the expression is highly variable, and I think that shows in the data that we saw with most of the kids, at least, not really having a bleeding or bruising phenotype that rose at least to the level that we'd expect. So I think you really need to jump whenever you have any data that suggests that there could be a problem in that family. So I think the diagnosis needs to be made. And I think the other important part is that we do need to establish baselines early and start monitoring. The bone marrow is a question. It's difficult. It's difficult for us. I mean, I'll put that right out there. We can't do a bone marrow on someone under a certain weight and under a certain age, to be honest. It's a little bit easier, I would say, is at a tertiary children's center or something of that nature who is a little bit more set up than we are to get that baseline bone marrow. But even there, it's difficult. And there are considerations about things such as anesthesia and the like in a very young child. If this was someone who had active leukemia, you wouldn't think twice about it. These aren't kids, but these aren't kids who have active leukemia. And so exposing them to risks needs to be balanced against the fact that about half won't get cancer. And certainly most people aren't getting cancer as a child. So I think our recommendation from that standpoint would be to establish the bone marrow biopsy as soon as it's generally considered safe and accessible. And that usually ends up being somewhere in like the 2 to 3 year range, probably closer to 3, unfortunately. And so far, we haven't seen that many cancers in that young of an age. It's just that it is a cumulative risk that piles up, and it piles up quickly in childhood. So you don't want to miss it. And then there's a couple reasons behind that. I think one is obviously to look for anything obvious that's coming out, but also just to establish a baseline. RUNX1 bone marrows don't look normal. That's sort of a given fact. And we know probably there is a good chunk of people in the past who have been transplanted or received our treatments with RUNX1 who didn't need it, but because we didn't really know what it was, they thought they had MDS or the like, and so they wanted to jump on it for however they did that. But still, having that baseline is helpful if there are changes. And then once you have that, but even before you have that baseline, and especially once you have it, to be doing blood work, CBC with differential 2 to 3 times a year, I think is a good place to look at it. There are a couple exceptions to the rule, but generally speaking, once someone was diagnosed with leukemia or other heme malignancies on our study, there was a suspicion for it even beforehand, even if it wasn't a clear diagnosis. So having those CBCs really, no one went into in other words, no one really goes into a bone marrow biopsy without a concern for it, especially if you're doing it more than once a year, if there's a reason you're doing it sooner. But I think that combination of having that bone marrow biopsy, the baseline in hand, doing it regularly to make sure look for changes, and that also kind of resets the baseline, and then having those CBCs every 3 to 4 months really, I think, sets you into a maintenance or a monitoring system that really is going to capture things early. And we don't have great data just because the ends aren't there. And, there are hopes to have new trials coming in for treating transplant and things like that to understand how that affects the outcome. But as a general rule of thumb, the sooner you catch it, the earlier you catch it, the better the likelihood of your outcomes are. Which is the way we explain this to patients. It's the way we're going to explain it to them. Where we're going to talk about it at the meeting next week is this isn't great news. We don't like to say that, oh, your kids are at risk. We hate saying that, to be honest. But this also is an opportunity to make sure that we fill this gap and that kids are getting the monitoring that they need, and to really establish an opportunity for them to really have the best chance at a good outcome compared to if we just go in blindly or if we just ignore the data. So I think those are really our 2 big recommendations, is to establish a diagnosis early, and then to really be doing that monitoring as early as possible with the bone marrow biopsies. In terms of intervals, right now we're saying once a year. That's probably more than we need. We just don't know the natural history. But our general gestalt has been you need to have a regular bone marrow biopsy, and we wouldn't start spacing them out until you had like 2 or 3 normal, stable ones under your belt. And that goes for kids as well as adults, and sort of how you have it. And then the third one, which is maybe a little bit more controversial, but probably not terribly so, is that we do recommend upfront transplant for all malignancies, regardless of whether they go into remission, as a consolidation process, because it is it both takes the possibility of a second malignancy, which we have seen completely independent, developing 2 leukemias in a lifespan if you give them enough time. But it also takes away it reduces the likelihood of relapse and things like that, which is a high concern in some of these leukemias that we are seeing from these patients.
Thank you, David. I appreciate you commenting on all that. All right. I know Nancy had asked the question about gaps, and then I filled the void with my question to David. But have people been thinking more about some of the gaps that they see? Or go ahead, Anipriya.
Yeah, I struggle with this question every day, like how to model inflammation accurately using murine model, right? Like we have tried multiple things and we really don't see what we have observed in patient bone marrow, like really high inflammation in like clean mouse facility. We haven't seen the impact of modeling one cytokine at a time. And I was just curious what people have done in their lab or what they think would mimic inflammation in mouse model, or modeling inflammation in mouse model to more accurately model what we see in the patients. So like, for example, for the context, like RUNX1 FPD, like RUNX mutated mice or RUNX flox mice with tattoo doesn't show all those allergies or immune disorders and things like that. We do see patterns, but nothing to the extent what patient experience. So I feel like we need something else beyond what we are doing, and that may impact how fast disease can expand in mouse model. Like we age the mice also, we haven't seen very robust myeloid phenotype or things like that. So I was just curious what people are thinking, how we can model using murine model, considering patient samples are highly precious and limited, and we can't do everything mechanistically using patient samples.
So, and I'll second your concern, because if we look at the cytokine profiling that you reported in patients and the ones that it has been done in mouse, they don't match. In general pathways, they are associated, but they're not matching. And if you look at the work that we published with the RUNX1 and 188Q, and you have to wait almost a year to get a phenotype. So, are we working in 2 clean facilities, and that is unlike what happens in human? That's a question that the RUNX community has talked about since, I don't know, at least since Nancy moved to UPenn, for example, that her phenotypes were changing. And we are actually doing something that one of my technicians took the leadership to do it, is that we are comparing, we have 2 facilities, one that is much cleaner than the other. And we are putting mice in the 2 of them, and wild types and with a RUNX mutation. And we are taking bone marrow fluid to see actually whether we see a difference in the cytokine profiling there. And if we do, we'll put the RUNX1 with a mutation and the delta allele to see whether the disease advances. That will explain why this is happening, but it will not solve the problem. We'll say that this is a big problem that we have with the mouse model. And so whether looking at inflammation in the mouse model is the best model for a human is a big question.
I don't know if we should be doing more in the rhesus macaque. I think it's going to be
rhesus and what?
Rhesus macaque models.
I wish it wasn't so expensive, and I wish
right,
it was right. And they're limited too, right?
And restricted, yes.
And then would not there be same issue because they also live in the controlled environment?
They do.
Yeah, but I think just the mice handle RUNX deficiency differently than humans.
Mm-hmm.
And I think, the limited data we have from Cindy does suggest that they're more closely model the human disease.
I don't know if any cytokine profiling is being done in macaques to see whether it's closer in the pathways and in the that are increased as a human compared to mouse.
Yeah, I don't know. I'll follow up with Cindy if she did that with the 2 macaques. I don't remember.
We
if she-
didn't, and
Oh.
regrettably, obviously the one died. But we are
Right.
actually, we still have the other one, and we were like, that one's just sitting there, kind of bored. We're actually going to do another testing on it sometime in the very near future. So, maybe we should add that to the list of samples to test.
Yeah,
I would imagine we would want to ideally look at marrow cytokines as opposed to peripheral blood, but that is what we were thinking of doing, is a bone marrow biopsy on the monkey. So
Probably have to do proteomics or something, because I don't think there is a cytokine panel specific to macaque, but maybe it's enough homology with human like
we've used one for our CAR T experiments, and
Okay.
it's worked well. But it's not. And you're right though. There are some gaps in it because there's non-homology, but there's a decent amount, especially for the big cytokines. But I can double check what's on that panel too.
You know, a related thing that I often struggle with is the phenotype in haploinsufficient mice, or even Lucio's mice, so mild, makes things so difficult to study. You know, are we being too much of purists to be doing everything in those models, or should we be really exploiting full knockouts more?
I think the hat is okay to have, but we do need full knockouts because it's like so mild, like as you said, and we do need to bring closer to what observed in human cells. So that's why, like initial experiment, for example, in our case, we did hat and then we shifted to more homozygous, because just like as you said, and others also, like after year-long waiting, it's just like very long experiments, and you don't even know, are you in like right CRI, right combination of mutations, right allele burden, and so on. It's like lots of questions.
Yeah, it's time and money, right?
Yeah, so now we have VAV-CRI, SCL-CRI, and mixed-CRI, all three going for tattoo. Because VAV-CRI phenotype in tattoo is so robust compared to SCL-CRI, which was a surprise, which makes sense now. VAV-CRI, like you don't induce in HSC, so compared to SCL-CRI. So I think there's lots of differences and challenges to model.
Mm-hmm. Kennedy, please go ahead.
Yeah, Katrine, there were two things that strike me from the presentations. The first one, the comment that short telomere is almost universal came to me as a very new piece of information. And short telomeres have received a lot of attention in a lot of other areas. There are lots of other groups, some of which that you mentioned maybe were interested. So it may be worthwhile asking the question, is there some way of expanding the story of short telomeres in FPD-ML with the other groups that are working? So that was number one.
Yeah.
Second was this story about MLL5. I thought that was very interesting, and given the fact that it's upregulated in AML and downregulation of that particular single compound protein seems to have an effect in changing the phenotype.
Yeah.
But that data was essentially from, if I remember correctly, from mouse models. It's worthwhile asking the question, how much does it apply to the human model? Because then it provides a single protein that, or a single gene that one can target. So that was the second part that I thought was very interesting from the discussions today. And these two areas have certainly not received much attention so far in the context of FPD AML.
Thank you, Kennedy. I wholeheartedly agree with you. I thought the MLL5 data it's just shocking to see such a reversal, right? When you knock it out.
And here's an admission. When I first saw those data and that Wenbin had chosen to study the one gene that was a dependency for even the R188Q heterozygote, I wouldn't have touched that with a 10-foot pole.
Yeah.
And yet he got enough. Again, some of the single areas.
Yeah, I didn't have that sophisticated thinking process. I thought it's just shared dependency.
Goodness.
When I had to do it.
And Wenbin, I know there are some drugs in development for targeting at least some relate, maybe not MLL5 directly, but at least H3K27 methyltransferases. Is that correct?
Yeah, so that's what Bobby tried. Yeah, Bobby tried the menin inhibitor.
Oh, the menin, right? That's right.
Yes. Actually, we have been playing around in the lab as well. We saw something different from Bobby and then Nancy. We saw that the menin inhibitor actually inhibit IQ knockout cell. A few times now. So I'm trying to validate that and trying to do a long-term like several week culture because menin inhibitor act slowly. Yeah, it's not a kinase inhibitor, but it's an epigenetic regulator. So we'll see. I mean, Nancy and I, discussed this how it works. It might, like RUNX1 probably somehow exclude the many complex from from the binding. So we'll see. The other thing is that we have been trying to develop a like protect for ML5. It turns out ML5 is a very poorly studied molecule. There's no well-known ligand for ML5. And we even work with a AI approach lab trying to screen like millions of potential small molecule for it. We couldn't get a very good target. There are a few, but very poor specificity. But on the other hand, for PROTAC, we probably don't need a high affinity ligand or high specificity. Even low specificity could be fine. If we target on other ML family, probably is fine. So we'll see. We're working on that.
And when we were testing the menin inhibitor against cells with a B-core mutation versus cells without.
Oh.
So it's totally different.
Okay.
Background. Yeah.
Mm-hmm.
I had one question. Did the patient who we heard from say that psoriasis is protective? Did you hear that, or did I miss? What was that about? Like,
I think you just said that she had
Her
it.
her mom had told her and her sisters that the people in their family who had psoriasis didn't develop blood cancer. And she felt like it was kind of a protective thing that her mom was saying because she didn't actually know
didn't
that.
think it was true.
But the mom thought it was true. I think she did, or she hoped it was true.
Well, I'm just thinking of it as a, there's microbiome and stuff like that go into skin integrity, and you could have some weird mechanism where psoriasis is protective. I think Elaine Fuchs had a really beautiful story in The Skin where psoriasis was protective from skin cancer, but
really?
atopic dermatitis was bad for skin cancer. So, these are, who knows if they're right or wrong, but it's interesting.
That is interesting.
Mm-hmm.
Well, we've reached. Oh, did someone else want to say something? I'm open to.
Yeah, I just had a comment since you were asking about things that maybe worth looking into more. This is anecdotal, but I remember from a meeting some patients last year at the meeting, and I forgot his name now, but he was telling us that he had the impression that in his family, the people who get blood cancer are younger and younger over the generations. So, and that fits, just to point this out, this fits somehow maybe with the telomere length. There's a speculation at this point, but I know for other telomere disorders, there is an effect over generations that telomeres become shorter and shorter, and then it becomes a more and more severe problem. So that is maybe something to look into, cross-generational analysis of telomere lengths, to see if there is really a shortening happening that leads to earlier onsets over generations.
I think that BC showed at least some data so far, right? Where she was trying to get at that question and by looking at the parents versus the children. And there wasn't, did I get this right, David? She didn't see trend?
It was a mixed bag, but there was a certain element of it where we did see it in the lymphocytes, which is maybe a little bit more convincing than the granulocytes, given the biology of telomeres in the two subclasses. So, but not enough to really make an argument that one way or the other that there is anticipation. It is regarding actual cancer. It is something we are looking at actively. We have a large, it sort of hinted at in the one chart where we showed the bimodal distribution of age of diagnoses. We have all the ones who are enrolled, but we do have a cohort that's, or not a cohort, it's not really say. We have pedigrees for most of our families, so we are trying to dig deep into that data. It's just difficult to do, and there's a lot of statistical considerations. So really the best way is to get more families directly enrolled, which is a challenge, especially since if you just go beyond a generation or two where the leukemia just is not something you survived, 2 generations ago, unfortunately. So we don't have those patients. But we are trying to look at it and to pull that apart. We have families where it really does seem to scream an anticipation, but we do have a couple families where it almost seems to argue the other way. So it's hard to say. It is difficult, but that is something that's in the back of our mind pretty much constantly when we look at this data.
Thank you. All right, I realize we've gone beyond our planned time together. I just want to thank everyone that stuck with us to the bitter end here, and thank you for the discussion. I always learn a lot, and it's really, really helpful as we think about next steps and how we chart the path forward from here. And I'm looking forward to seeing all of you in person, hopefully sooner than May, but absolutely in May. So please make sure that you register. And again, thanks to all of you for all the work you do. It's really powerful. And I know we have a whole patient community that's very, very grateful. So, and I'm looking forward to sharing the key takeaways with the patient community with Nancy next week. So we'll report back on what we learned from them all as well.
Wonderful.
Take
Thank
care.
you very much.
Thanks, everyone.
That's a great meeting.
Thanks, everyone.
Bye.